Hello. How are you doing? Can you hear me? If you can hear me, type on the. Oh, okay. So my voice is very clear. Ah, Sarah, how are you doing? Okay. Good evening. Good evening. How many minutes do we have? Okay, we have 1 minute. So let me see how many people I have here. One, two, three. Please, if you are here, I need you to type in the box. This will be a very fast-paced review, and it will be very interactive. So we have 11 people here, and I need up to 20 people to start up. So please, if you are here, just type in the box, the chat box. You will comment, you will communicate with me more from the chat box, all right? One, two, three. Okay. Good evening. One, two, three, four, five, six. Good evening. Mm. Hello. Can you all see my screen? Yeah, okay, good. Sorry, I'm using this for the first time. I needed it to record so that the other people that are not here will be able to use this video for today. Let me give them 5 minutes for us to start. So this is 9:01. We'll start by 9:05, please. So that I'll be sure everybody's here before I start. So for us to for me to be able to understand, what are the areas that's more troublesome for you guys? Can you type in the box? Is there any area that you don't understand very well you want me to pay much more attention to? Because today we are supposed to make a review for the three topics that you'll be writing an exam on, which is tissues, genetics, and skin. So chapter 6. Okay, chapter 6 is skin. Skin, right? Okay. Okay, good. Good. Is there any other area that you guys are having issues with in the other two chapters? Chapter 5. Okay. Chapter 5 is tissues. Okay. Chapter 4, genetics. Okay. That's fine. Chapter 5 as well. Okay. That's fine. That's good. So the way I want this thing to happen is I will make a very fast, because we don't have much time. I want this thing, this our review to be like 2 to 3 hours, so that I don't bore you out. You know. So, but all of you has to be actively engaged. I'll be putting questions in the box. That's the rule, please. I'll put questions in the chat. you look at it and you tell me what your answer is. All right. Either you type your answer or you type it A, B, C, D, and E. Okay. And sometimes some of the answers may not come with options. So I will need you to give me your answer without even option. And you will have to do it very fast so that I can show you, some of the questions that the way they structure the questions that comes out in your exam. That's the idea of the entire thing. So we are not here to just start having a new class altogether. No, because if I want to take these 3 topics as a class, it will touch an area we should be familiar with. Hey, you're a politician. You should be familiar with some areas. So as an advice, I want you guys to pay much more attention to genetics and to tissues. You understand? Because that's usually the area where most of your questions comes from. Same thing with skin, but skin to some extent, all right? Because your professors are always more focused on genetics. Okay, this is 905. So I will give a very short recap before I start putting questions. So I need you guys to be very, very active, all right? And those questions, I won't leave it for a lot of time, maybe 30 seconds. I need you to give me an answer in 30 seconds. If I get 10 people, if 10 people picks an answer, I'll move, all right? So that's how we are going to do it. Then I can explain some of the concepts for you. All of us know what genetics is about, so I don't need to start explaining genetics anymore. But it's very important for us to study genetics because of some of these diseases mentioned here. All right. Now, what you need to know is the components of the DNA. DNA. Remember, DNA is a polymer of what? Nucleic acid. So you should be more interested in what is nucleic acid or nucleotides rather. DNA and nucleic acids are polymers, right? Made up of what? A lot of monomers. But the major unit is nucleotides. And your exam, what they will be asking you on is nucleotides. So the question you need to understand is what is nucleotides? And nucleotides are, according to what you have here, they consist of sugar, phosphate group, and nitrogenous bases. Sugar, phosphate group, and nitrogenous bases. They will test you on each of those components apart from phosphate group. You understand? So the phosphate is the backbone. We already know that. But they'll test you on sugar. Which type of sugar does DNA have and which type of sugar does RNA have? That's what you should be thinking about. Which type of nitrogenous base do we have in DNA and how does it differ with the nitrogenous base we have in RNA? What are even nitrogenous bases? You understand? What are the classifications? These are the things you need to put your head to understand. All right. If you look at this particular page, you see this is purines, this is pyrimidines. Purines, adenine and guanine, pyrimidine, cytosine, thymine, and uracil. Uracil is usually found in what? RNA. So anytime you see you are talking about RNA. All right? Extremely important. So cytosine, thymine, that's for DNA. Adenine, guanine can be for DNA and RNA. All right? Very, very important. So for DNA bases, we have ATCG. And remember, DNA basis comes in threes. In threes. That's why we call them triplets. So put that in your head because there's a question about it. Now, remember, we mentioned that there are purines and pyrimidines. So when you see purines, they are double bonds. I always remember them by remembering that, purines is small compared to pyrimidines. It's small, but it's large, right? It's small, but it has double bonds. Pyrimidines is large, but just one bond. It's as simple as that. So if you look at this diagram here, you see this is one ring, this is another ring. One ring, another ring. All right? One ring, another. So these 2 rings, that's double rings. So and if you see a diagram like this in your exam, maybe your lab exam, for example, you should know that this is what? Purines, because it has double ring. They may not be testing you on, is it adenine or is it guanine? That is biochemistry. When I take biochemistry class, I will take you deeper, with the structures. But what you need to know is, most importantly, right now, is the rings, all right? Then this is purines, one ring, cytosine, thymine, and uracil. Extremely important. So this complex is known as what? Nucleotide. Phosphate, ribose, deoxyribose sugar, and what? And nitrogenous base. 1, 2, 3. So the 3 of them makes up what? Nucleotide, as we mentioned here. And DNA and other nucleic acids are polymers of what? Nucleotides. In humans, there are 46 DNAs, DNA molecules, that are chromosomes. Very, very important. You need to be able to understand this. And DNA is double helix. Double helix. Double helix, 1, 2, 1, 2, double helix, one helix, another helix. Extremely important for you to know. Now you need to understand what genes are. They are segments of the DNA coding for the synthesis of a specific protein. For people that are in my class, I gave you stories that you should be able to understand what the DNA is. All right? What the gene is. I'm not going to continue discussing it here because it will take my time. Now, you need to know the difference between gene and genome. Gene are segments of DNA coding for the synthesis of specific protein. Why genome are all the genes somebody has? They will ask you this question in the exam, so pay attention to it. I'll give you some questions surrounding it. Now, we have chromatin. We have chromatin. They are DNA materials that are complex with proteins. It's like they are tied, all right, with proteins. Very important for you to know the meaning of chromatin. And this is the process of chromatin structure. This is the DNA, all right? And this is how, it's being tied, all right? So look at it at your leisure. Then there's something called histones. Histones are those proteins that are crucial for DNA packing. They package the DNA, all right? So they help to hold the DNA in place. Extremely important that you should understand this as well, because they will ask you a question on histones. Now, another thing you need to know is that genes can be turned off and turned on. For example, in the lab, we are working on an organism, Pseudomonas putida, and we are knocking off some of its gene and knocking on others. Do you understand? So genes can be turned off or turned on depending on its importance at a particular time. Okay. So so for your lab class, you need to understand what kinetochores are. They are protein plaques that are on each side of the centromere. You know what a plaque is? A plaque. Is it called plaque? Okay, plaque. Now, in when you discuss physiology of the heart, all right, pathology of the heart rather, you will discuss about a plaque falling off and causing thrombosis, all right? So that's when you understand what plaque is. So plaque is usually something that is attached that could fall off at any time. You understand? So you need to understand what kinetochores are because you will see it in your exam. So if you look at this diagram, they may give you this diagram, they may point to this and tell you what is this. This is kinetochore. And this area is known as the centromere. This place is known as the centromere. And this has two sister chromatids. Also very important for you to understand. Now, you need to know the difference between RNA and DNA. Specifically, you need to know the major differences. What are the differences? I'll tell you now straight up. RNAs, their sugars are ribose. DNAs, their sugars are deoxyribose. Does it make sense? RNA, their sugars are ribose. DNA, their sugars are what? Deoxyribose. You should remember that. All right. Now, in terms of the nitrogenous base in RNA, RNA has what? Uracil as one of its nitrogenous base. DNA does not have uracil. You understand? Instead, it's replaced by what? Thiamine. I'll give you questions around that. Let me see what else you need to know. This is definition of gene. The most important thing you need to know is what is gene. All right. It's just the part that contains the major information that you need for an organism to multiply. It's as simple as that. Here they mentioned that gene are information containing segments of the DNA that codes for production of molecule of RNA that plays a role in synthesizing one or more proteins. So you just have that concept in your head, all right? Now, genetic code are the information that is contained in DNA. That's what you should know. A system that enables 4 nucleotides to code for amino acid sequences of all proteins. That's genetic code. Very important. So we have base triplets. I told you they may ask you what is base triplets. Base triplets is just 3 base sequence in a messenger RNA. Very, very important for you to know this. Now we have something we call stop. We have start. They may ask you one of the following is an example of stop, or one of the following is not an example of what stop. This one you have to put it to your head. There is no circumventing it. Extremely important. So stop codons, UAG or UGA or UAA. Extremely important. Once any of them is found in the sequence, what will happen is that the entire process will stop. And start codon is AUG. That one is easy. AUG. So please, you have to put it in your head right now. What is the examples of start codon and the example of what? Stop codon. Extremely important. You definitely will see it in your exam. I will give you a question on that right now. Now, protein synthesis is very straightforward. All right. We have replication, we have transcription, and we have what? Translation. That's the much I can say about it for now. All right. Remember, we have DNA polymerase and RNA polymerase. DNA polymerase does what? Replicates. DNA polymerase does what? Replicates. So first of all, the helicase what? Breaks everything open. Then each arm of the DNA, you know, it's double arms, right? Each arm of the DNA is now replicated by the DNA polymerase. When it's now replicated by the DNA polymerase, that replicated part will now be transcribed to messenger RNA. That is called what? Transcription, no longer replication. Replication is when DNA is, photocopied. But transcription is when, that code is written down in form of what? Messenger RNA. Extremely important that you know this. And what helps in transcription? RNA polymerase. They will ask you. DNA polymerase for replication, RNA polymerase for what? Transcription. This, as I'm talking, if you have any question, you can just put it down there. All right. And this is the correct. They will give you a question on this particular place to make a corresponding messenger RNA. I'll give you something like that right now so that all of you will try it. All right. Then we have alternate splicing. All right. That's variation in ways exams are spliced, allowing for variety of proteins. All right. How to use a certain particular code, particular genetic code to create different proteins. That's what we know as alternative splicing. You understand? Very straightforward. This is alternative splicing. So this is transcription. You see this transcription? Then this is splicing. And this is translation. But remember, before that, the DNA must have been what? Replicated. So replication happens here, transcription, splicing, and what? Translation. They will give you this diagram and you need to understand, especially, they can give you this diagram in the lab, all right? In the lab. If they point to here, you know that this is what? Replication. They will ask you what process happens here. This is replication. What process happens here? You understand? This is what? Transcription. Why? Because you see pre-mRNA. So DNA to mRNA is what? Transcription. That's how you know that. Because somebody will ask me, oh, but how do I know, Professor? How do I know that, okay, this is transcription? That is exactly, immediately you see pre-mRNA. As far as you see mRNA there, that is transcription. Moving from DNA to RNA. But DNA to DNA is replication. Now, when you create this transcription, they splice it, they cut it into different segments. That is what splicing. Now, they use this guy to make this particular type of protein, use this mRNA too to make this particular type of protein, and this guy to make this particular type of protein. All right? Now, the three, this is called what? Translation. Translation is when they make the protein proper. So all this talks about our discussions in translation, which I will give you a question for you to understand what I'm talking about. And after translation, what happens? There is packaging and exporting. All right? Where does it happen? Usually, transcription or translation rather happens where? In the cytoplasm. It can happen in ribosomes that are circulating around. All right? It can be ribosomes that are just moving freely, or it can happen on ribosome that is found where on rough endoplasmic reticulum. What makes it rough? It is rough because it has what? Ribosomes attached to it, just like sand. If you touch it, it's very, very rough. All right. So you should be able to understand the question that they're asking you. You need to be able to understand it properly. And also, they will ask you a question on chaperone proteins. They're just older proteins. Like most of you are nurses, or everybody here is a nurse, probably. Now, in the hospital, you have somebody that will escort you to see a patient. That's a chaperone. That's exactly what these guys do. All right. So chaperone proteins are usually old proteins that tells new protein. No, this is how you have to fold. This is how you have to fold. Because if a protein folds wrongly, there'll be a problem. It becomes pathology. You understand? And remember, what are we discussing? We are discussing what? Physiology. And physiology is what? The right thing. You see how I'm moving my hand? This is the right thing. A situation whereby my hand is like this and I'm not able to move it, it becomes what? Pathology. Because the physiology is that I should be moving it. It's as simple as that. Physiology, I should see. If I'm not able to see, it becomes what? Pathology. So remember, whatever we are doing is the real thing. So in physiology exam, they hardly ask you pathology questions. That's another thing you have to put in your head. They hardly ask you pathology questions. right? So when the proteins are processed here, this is the rough endoplasmic ribosomes in the rough endoplasmic reticulum. They are packaged where? In the Golgi complex. So the Golgi packages them and send them out of the cell through what? Exocytosis. All right? Extremely important that you know this. So this is everything about the Golgi for its modification arrangements and all whatnot. Sorry, I'm moving very fast so that I can we can start pouring out questions. These are exactly things that we've already discussed. Now, a second part of this discussion for genetics that you must know is what? Cell cycle. You must know what? Cell cycle. Now, using this diagram, I love this diagram a lot. You understand? They can give you this diagram in your lab, or it can still come out in your normal lecture exams. All right. Now there are three phases. How many phases do we have? Three. One, two. Cytokinesis is actually its own part, and three, interface. They can tell you to arrange this thing. All right. First of all, you need to know that this is G1, G2, G1S, and G2. This 1, 2, 3 are found in interface. Interface is resting. And when they are resting, that is when DNA is replicated. Now, they may ask you a question. Where is DNA replication? Where does it occur? That's one question they can ask you. Where does DNA replication occur? Where does DNA replication occur? They will not start giving you the options that you are familiar with. Instead, they will give you options like this, like mitotic phase, G1, S, G2. So you need to know that this is S phase. Very, very important. So we have G1, S, G2. Then we now have preparatory phase. That is prophase, P for P. We have metaphase, meta, M for middle. So everything line up in the middle according to your height. Then we have what? Anaphase. If they ask you, where does the division of the chromosome start? Anaphase. That's where they start division. Where do they line up according in the middle? That is what? Metaphase. So they start dividing in anaphase. Then they move to the opposite pole in the telophase. And after that, the cell breaks up itself where? In the cytokinesis. After that, the cell rests. That is in interphase. It's as simple as that. Honestly, that's everything you need to know in mitosis. All right, I'll give you some questions. Also, you need to know this diagram extremely well because this diagram might come out in your exam. They may bring this out for you or they may bring this out for you. You understand? This is prophase. Everything is still normal. If you see all of them are already aligned in the middle, that is what? Metaphase, M for M. Now, if you see here, they have started breaking, that is anaphase. They start moving to the individual poles, that is telophase. Then here they break into two cells, that is what? Cytokinesis. And they start resting. All right. So you need to know that. Then you need to know the checkpoints. We have a start checkpoints. At G1, we have a checkpoint at G2M. All right. And we have a third checkpoint here. A third checkpoint at the transition from metaphase to what? Anaphase. These are the three checkpoints. And you need to know them. That's why they are colored. All right. Then you need to know about the molecular timers, which are the cyclins and CDKs, which is cyclin-dependent what? Kinases. Cyclin-dependent kinases. And this cyclin-dependent kinases usually match with cyclins to form what? To form cyclin-dependent kinases complex. This is the one that do the control properly. All right. I'm not going to spend much time on it because we have other things to do. Then you need to know about hereditary, the meaning of hereditary, the meaning of karyotype. Karyotype is just a chart of 46 chromosomes. All right. This is, if they give you this diagram in your exam, you tell them that this is karyotype or karyotyping. All right. If you look at it, this is chromosomes from 1 to 22. So we have 22 pairs. Then we have of body chromosomes. Then we have one pair of sex chromosome. So for a female, biological female, the person we have this 22 and this excess. Then for a biological male, you have this 22 and what? This particular one. So you need to be able to read karyotyping. And we have diploid, we have haploid. All right. We have diploid and we have haploid. Diploid describes the cell with 23 pairs of what? Chromosome. That's somatic cell. So if I cut out this cell from my body right now, here, this body cell, if I cut it out, this tissue, and bring out one of the cell will be what? Diploid. So it will be 46. That's 23 pairs. All right? But if I bring a sperm cell or an egg cell from a woman, that sperm cell will be 23 unpaired. That's what we call what? Haploid number. Just 1, 2, 3 to 23. But if it's body cell, it will be 1, 2, 3 to what? 46. That's 23 pairs. You get the mathematics. So, but haploid is just sex cell and what? Sex cells, that's egg and what? Sperm. Simple. That's what haploid is talking about. So don't get it confused. It's very, very important that you understand what they are talking about. All right. Then you need to know what the locus is. We already discussed this. Just the location on a particular gene or a chromosome. Then we have alleles. We have dominant alleles and recessive alleles. I'll give you a question on this. Then you need to know what genotype is. That's alleles of an individual, all right, that have to depict for a particular trait. Then we need to know what phenotype is. Phenotype is what you are seeing. You see how handsome I am right now? That is phenotypic appearance. Somebody is laughing over there. That's phenotypic appearance, right? It is my genes that make me as handsome as this. But you don't see the genotype. What you see is what? The phenotype. What is expressed, all right? Why Punnett square is just the box that, you match genes, all right, to be able to predict a phenotype. You got it? Okay. Then you need to know what gene pool is. It's just genetic makeup of the whole population. You understand? You have to know what codominance is, what incomplete dominance is. I will explain it with the equation. Now, you need to understand polygenic inheritance and pleiotropy. Polygenic inheritance is genes are 2 or more loci contributing to a single phenotype. I am dark like this because there are multiple genes, maybe like 3, 4, 5 different genes on different locus that made me dark. So meaning that skin color is what? Is a polygenic inheritance. That's not what we are talking about. One gene can never make you this, can never determine your complexion. Same thing with eye color. All right. So if they ask you what is the example of polygenic inheritance, or they may tell you one of the following is not an example of polygenic inheritance. You need to know that the major polygenic inheritance are what? Eye color and what? Skin color. Just put that in your head. Same thing with pleiotropy. Pleiotropy is just an opposite definition of polygenic inheritance. Remember, polygenic inheritance, I said when 1, 2, 3, 4 genes are coding for this particular thing. But pleiotropy is when one gene has different physical expressions. For example, in pathology of acaptonuria, all right, it leads to different things. Let me give you let me show you what it leads to. Look at it. This is gene 1, gene 2, gene 3. Okay, this is polygenic inheritance. 1, 2, 3. They are coding for eye color. Now, if you look at pleiotropy, this is one gene. This one gene causes a darkened skin. This one gene leads to darkened sclera, and this one gene leads to what? Dark urine. This is the dark urine. This is the normal urine here, and this is the dark urine. All right. So this is pleiotropy. Why this is polygenic inheritance? So they are just opposites. You need to understand this because it can come out in the exam. All right. Then we have sex-linked traits. We have recessive color blindness, allele on X, and no gene locus for traits on Y. That is describing sex linkage. We'll discuss that with the equation. I prefer us to discuss that with the equation. Let me see what else we need to understand. Cancer is important, but I'm not going to discuss cancer tonight, all right? Read it at your leisure because I'm discussing physiology. I don't want to discuss pathology, all right? If not, these are just types of cancer. Very important for you to understand. Okay, that is it. Is there somebody, if you don't understand something, tell me before I start giving you a lot of questions right now. Anybody confused? No? Okay. So I want you guys to, so please, we'll have to hasten up. If immediately we are up to 25 here, or 24. Immediately I get 10 people commenting. Somebody say 1 second. Okay, you want to ask something? If you have a question, just type. Oh, you say 1 second to answer? Okay, I agree because we really need to be very fast. Sometimes you think 3 hours is big. It's not big. And your exam, you guys are writing exam soon. It's very helpful. Thank you. You're welcome. Okay, so let me start from the simplest question. Let me start from the simplest question. If you are given this question, DNA is a polymer of dash, which consists of a sugar phosphate group and dash. Forget about the blank, please. And dash. Okay, the options are, let me see. So these are the options. Yeah, the options: amino acids and nucleotides. Nucleic acids and nucleotides and nitrogenous base, nucleotides and nucleic acid, proteins and nucleotide. Let me see. Somebody said nucleotide and nitrogenous base. Nucleotide and nitrogenous base. Nucleotide and okay. So this is a giveaway. Everybody knows this. I assume that all of you understand this. So we move. Because if you look at the slide, if you look at the slide I discussed with you initially, you will understand what I'm talking about. Okay, so I'm going to give you this. I'm going to give you this. And you tell me the answer. I'm going to give you this. So this is the question. This is the question. I'll be posting the answer one after the other, because I see that if I post it together, it's not differentiated. So this is A. That is A. Hold on. I'll tell you when I'm done. That's A. This is B. This is C. This is D. And this is E. Okay, fastest finger. We are 25 here. I need everybody to make attempts. That's A, B, C, D, E. Just put any of the options. Okay, we have number 1. Okay, let's keep going. Let's keep going. Let's keep going. Let's keep going. Don't pick what everybody is picking. No. Make sure you read the question properly. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So you see, this is the kind of question that they will ask you. All right. Yeah, thank you. Which of these is the correct sequence? So you see, from carbon to nitrogenous base to nucleotide, then to DNA and to chromatin. Extremely important. All of you are correct. Everybody is correct. And thank you for being very engaged. All right. Thank you for being very engaged. I will give you perfect. This is a very good question. This is a complete. Thank you, guys. Thank you. You guys did well. Okay, so let's go again. But this one, I'm not going to give you the right answer. I'm not going to give you option. You have to type the answer for me. All right. And then I will tell you who's correct and who is not correct. This is the question. Oh, yeah. Let's go. Let's go. Let me see who understands complementary base pairing. Let's go. Let's go. Let's go. I need everybody to attempt this question. Everybody. Feel free to get put something wrong. It's fine. All right. Don't do what everybody is doing if you don't understand it. Okay. Okay. I'm seeing. Somebody said chicken. Yeah, very funny. Somebody typed C. No, don't type C because I didn't give the option. I want you to type the answer by yourselves. Somebody say you guys are G's with typing. People are ready for this exam. I am very, very impressed. Okay, so we have a lot of answers. Okay, let's see. So T, good. You guys are doing well. So we remember they said DNA base sequence to what? DNA strand. So T, we go with A. That's correct. For people that got it right, T goes with, thymine goes with adenine. The next one is G. Guanine goes with cytosine. That's correct. Somebody said we are not ready, we are not fast. I don't believe you. You guys are answering this thing so fast. I'm very impressed. And you are getting it correctly. So the answer is, let me type it. The answer is thymine goes with adenine. Guanine goes with cytosine. Then cytosine goes with guanine. Cytosine goes with guanine. And adenine goes with thymine. And thymine goes with adenine. So this is the right answer. Yes. Yes. Let me see. Up under the tree. Oh. ACGGTA. ACGGTA. Okay. Okay. I'm impressed. Okay, let's go. Let's move faster. So I don't need to put options anymore then. I see that you guys are very good and I love it. It makes this faster for me. So this is okay, you know what? If I give you the question, I can just read out the option instead of me typing it one after the other. You just put your answer. So you have to pay attention. A dash contains the necessary information for the production of a molecule of RNA. Okay, let me see. Okay, so we have genome as A, gene as B, nucleotide as C, genetic code as D, and codon as E. Okay, let me see. No, I will just read out the options so that it will be faster. Wow, wow. Okay. Let me see. Okay. I need to get the 10 people to get it correctly for me to move. I have 1, 2, 3, 4, 5, 6, 7, 8. 2 more people to go. Come on, guys. Are you sleeping? Please don't sleep on me. Okay, so we have 10. What is okay? So this, guys, tell me what is the answer to this? Very fast. Let me see who gets it first. Very fast. Oh, somebody said G. No. We've moved beyond that step. We are now in the next question. Okay. Somebody said genome. Let me see if she's correct. Somebody said genome again. I need 10 people to attempt it. Options: A, centromere; B, chromatid; C, karyotype; D, phenotype; E, the genome. Let me see. 1, 2, 3, 4, 5, 6. So nobody is picking phenotype here. Okay, so we have 10 people. Thank you. The answer is genome. Genome is the set. So the DNA, 23 chromosomes, they are referred to as genome. Okay. Let me see. Let's go to something more difficult. Something more difficult. Okay, genome. Okay, so let's go. This question. Next one. Are you guys ready? Yeah, you are ready. Okay, let's go to this. Next question. Bam. I'm not giving options. Nah. I'm not giving options. So tell me, what is the answer? There has to be 2 answers, all right? So I need you to type the first one and the second one together so that we'll be able to understand where you stand. Okay. Somebody say answer choices. Okay, fine. Transcription, translation is A. Translation, transcription is B. Replication, translation is C. Duplication, transcription is D. Translation, transcription is E. Transcription, okay. This happens in nucleus. Translation is cytosol. Okay. Let me see. How many people have attempted? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, perfect. You guys are correct. Yes. So DNA to RNA is transcription. All right. Using information contained in RNA to make protein is what? Translation. Simple. Check it out. Okay. Now let me ask you this question. Please pay attention, because if I put the question, I will just read out the option. I don't need to. I don't want to be posting it one after the other. So this is the question, and everybody, please, I need you to be engaged in this. I'm very proud of you guys. Honestly, like I'm happy when everybody's getting it very fast. So which of the following is not directly involved in translation? All right. Which of the following means I have to give you options. 1 is DNA, 2 is mRNA, 3 is tRNA, 4 is rRNA, 5 is ribosome. One is DNA, 2 is mRNA, 3, that's C is tRNA, 4 is rRNA, 5 is ribosome. Okay, okay, this is good. This is good. You guys are really doing exercise though. Let me tell you, I know. Okay, somebody said something else. It's interesting. Okay, so get the double helix out of here. I have to. I have to. But you know, like somebody mentioned ribosome. No, the answer is DNA among all of them. Ribosome is actually involved. All right, ribosome is involved. You say which is that not directly involved in translation. Anything that if you see the other options like tRNA, we mentioned that tRNA is the one that brings the groceries that's amino acids. Then the messenger RNA is the one that brings the code. Then we have the ribosomal RNA is the chief chef. All right. And right, all of them happens in the kitchen, which is the ribosome. Make sense? But DNA is in the library. Remember, DNA is the golden chef book that is in the library, which is the nucleus. It's not in the cytoplasm. All right. And translation happens only where in the cytoplasm, not in the nucleus. The only thing that happens in the nucleus is the replication and what? Transcription. So please, somebody mentioned, because somebody got it a little wrong. The person said ribosome. The answer is what? DNA. DNA is the one that is not involved. Ribosome is the kitchen where translation happens. All right. Thank you. Okay. No. This one is also a giveaway. No, this is a giveaway. This is a very big giveaway, but I'll still give it away. All right. So, but this one will not take you 1 second. All right, let's go. Let's go. Let's go. Let's go. Here is my people. Oh, okay, okay, okay. Do you need option for that one also? No, this is giveaway. I'm not giving you option. I'm not giving you option. Yes, you said it. You actually said it. How do you know that I'll bring it out next? Okay, probably because I've said it a lot. Yeah, so she's very fast. Yes. Okay, yes, you guys are correct. Transcription occurs where? Transcription. Yeah, you got the answer is what? Nucleus and cytoplasm. Transcription occurs in nucleus and translation occurs in cytoplasm. I just mentioned it. All right. Please, if you see anything similar to it in the exam, you cannot afford to fail it. Please don't. Don't. But if you get it wrong, if they ask you who taught you, please don't say it's Nchebe. Okay, let me give you this. Okay, let's see. Let's see. Dash are turned on or off by regulatory proteins in accordance with the changing needs for the dash they encode. Let me give you options. No, let me give you option in this one. Let me make life easy for you. Dash are turned on or off. So there are two answers. A is enzymes products. B is ribosome proteins. C is gene proteins. E is proteins gene. Okay, that's D. Then E is gene ribosome. Answer. A, enzyme products. B, ribosome proteins. C, genes proteins. D, proteins genes. E, genes ribosomes. Answer. Okay, let me see. Come on, guys. Come on. We're up to 31. So I need a lot of answers right now. Come on, let's go, guys. Let's go. Genes, proteins, genes, proteins, genes, proteins, genes, proteins, C. Genes. You guys are correct. You guys are correct. Genes can be turned on or off by regulatory proteins when they want to produce what? Proteins. They can be turned on or off, when they want to produce proteins. Okay. Let's go to something more difficult. The truth of the matter is that if you understand genetics very well, nothing will be more difficult for you, because all these questions are very difficult questions, Somebody said, I might not say no. Thank God he didn't say it before I collapse this evening. Please, I'm not eating. You guys should not make me collapse. Okay, let me show you this. Come on. Somebody should impress me. Okay, I'll give you options. A is splicing. B is post-transcriptional. C is post-translational. D is polyribosomal. And E is secretory. Splicing, post-transcriptional, post-translation, now, polyribosomal, and secretory. Somebody's asking me, is it post-translation? Post-translational, that's good. Let me see if somebody will tell me no. You talked about it too much. Yes, because that question, everybody loves that question, not just me. Okay, you guys are correct. The answer is post-translational. But look at this question. Everybody. Look at this question. Since there are no genes coding for carbohydrates, nucleic acid, or lipids, how do cells produce them? Options. This is A. Hold on, let me give you the options before you start picking. This is B. This is, just pay attention, so that you get it right. I need you to get this answer right. This is C. This is D. And this is E. Are you sure it's not E? Okay, let me see how many answers I get here. I get 1, 2, 3, 4, 5, 6, 7, 8. Okay, let me see who gets it wrong. Okay, you guys are correct. Okay, you guys are perfectly correct. Thank you so much. So the answer is A, as you said. You correctly said the answer is A. The answer is A. Very, very important that you know that. Yeah. And A is enzymes are encoded by genes synthesizing genes that synthesize these products. So usually products like lipids, for example, carbohydrate, they don't have genes for them, right? But instead, the body creates genes for enzymes. The enzymes will now, create genes. The enzymes will now, act on the carbohydrates. That's how it works. All right. Unlike protein. Okay, let's see this. Let me see if you guys are following. Try this. I thought I've posted it. Oh, I've not. Hold on. Oh, I don't know that there's maximum of. Okay, try this, guys. Try this. Okay, so I'll give you the pathway. Relax. Before you pick answers, just look at the question. I need you to look at the question and understand it. Look at the question and understand it. Look at the question very well and understand it. It's getting harder, I know. I understand. But look at the question at first and understand it. Now, let me post the options. So just hold on. Let me post 4 options. This is 1, A. Hold on, guys. This is B. This is C. This is D. Then this is E. Incoming, missile. So this is E. Okay, guys, let's go there. You were able to radioactively tag and thereby trace an amino acid that is used to make insulin, a hormone that will be exported out, you know, blah, blah, blah. Pathway of the tagged amino acid would be what? Let's see. Somebody say B and C. Okay. I love the fact that you're choosing 2 answers. Come on, we have up to 25 people here. I need everybody. Don't think you will be wrong. Don't get intimidated by your classmates, please, or your schoolmates, or your setmates. Try to make an attempt. Even if your own is the odd one out, you may be right. Okay, let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So the answer is actually B. All right. You guys are correct. You guys are awesome. Honestly, I'm shocked. So usually free ribosome, cytosol, rough endoplasmic reticulum rather, Golgi apparatus, Golgi vesicle, then they push it out through exocytosis out of the cell. That's what they're asking you, and it's exactly what you discussed. All right, that's after creating of the proteins. Very, very important. Okay. Now, this is the simplest question you may be able to see. This is the simplest question. I'm posting it now. So, and I will not give you the option. I won't even read the option for this particular one because it's too cheap. It is too cheap. If this question is dollars, it will be 50 cents. Okay, try this. So B, yes, you guys are correct. Somebody say B and C. Please remove the C. Throw it away. Okay, let's see. Let's see. Wow. People are getting good figures. Let's see. Some options 20, 51, 102, 153. $51 multiplied by 3. Yes. Okay. Yeah. I will explain it. Yeah. You all of you are correct. It's actually 153. All right. So what you need to know, is that gene coding. Remember I mentioned that gene coding is what? Triplets. 3. They come in pairs. Like AUG. Do you understand? So you see AUG, you see UUA, you know, that kind of thing. 1, 2, 3, 1, 2, 3, 1, 2, 3. So they're asking you, gene coding for a polypeptide is made up of 51 amino acids. You understand? What is the minimum? If it's made up of 51 amino acids, that means it will be 3 base pairs, each of them, each of those amino acids, 3 base pairs. All right. So if you multiply 51 by 3, it will give you 153. It's as simple as that. So if you just remember the triplets in your head, because you know the nucleotide sequence is triplets. You know? Like me, I want to give birth to a triplet. So you should know that the template for transcription is the molecule that carries what the base triplets. And that's how you are able to get that question. The answer to that question. So please remember it. Okay, let me see. Oh, perfect. Perfect. No, it's not mathematics. It's just common sense. It's not mathematics, honestly. Like, don't worry, you're done with maths. Okay, look at this. Look at this. Look at this. Talk to me, somebody. I'm not giving you option. And remember, this is cell cycle they are talking about. Okay, fine. Let me give you option. We have a S phase, G1, G2, M, and G0. The options are S phase, G1, G2, M, and G0. Talk to me, somebody. Let me see. 1, 2, 3, 4, 5, 6, 7, 8. Oh, we are done with by 2 people. Where are they running to? Are they tired of me already? Please don't get tired of me. Oh. You are stuck with me. Okay, yes. So the answer is actually G. Actually S rather. Pay attention. Remember, if you see the options I gave you S, G1, G2, M, and G0. The answer is S because that is replication. If you check in S, what happens in S is DNA replication. And DNA polymerase is what leads to DNA replication. I don't know if it makes sense to you guys. That's the kind of question they were asking. This is a very straightforward question, but it's a little tricky. It's a little tricky. All right. So you need to, if you notice what we discussed here, look at it. If you look at this diagram, see what we discussed here. This is S phase. This S phase is what? DNA replication. And DNA replication is caused by what? By DNA polymerase. That's what they're asking you. Thank you. You guys are the best. Okay, let's see. So we have 31 people here. I'm expecting 30 people to make this attempt this. Give me the answer to this. Do you need options? Okay, fine. I'll give you options. We have interphase A, telophase B, cytokinesis C, prophase D. Answer. Okay. The answer, okay, yeah, you guys are correct. You guys are very correct. So G1S, G2 phases of the cell cycle are collectively known as what? Interphase. Very straightforward. These are the straightforward ones. So now let me give you this one. Let me see if you can reason it out. Very interesting. I think why are the interesting questions coming at the end? Because I'll soon be done with genetics. Look at this question, guys. Option is S phase, telophase, metaphase, prophase, interphase. S phase, telophase, metaphase, prophase, interphase. Answer. Oh, okay, you guys are correct. The answer is what? Telophase, please. The answer is telophase. Oh, look at this very important question. Look at this very, very important. Oh, yeah, ask your question. You have a question, just type your question for me, all right? I will treat it. Please, anybody that has something that is confusing you, this is a communicative environment. Just type your question in the box. I will answer it, all right? So as you're typing your question, I'm asking my question as well. Okay. So the next question to attempt. Dash opens up one segment of the DNA helix during replication. Then do replication. Dash adds complementary free nucleotides to the exposed DNA strand. Answer. Please, the person that has a question, feel free to ask your question. Please, just type your question. But every other person should attempt this question. No, the options is supposed to be 2. It's supposed to be 2. Okay, options. I'll give you options. A, DNA ligase and DNA helicase. B, DNA helicase and DNA polymerase. C, DNA ligase and DNA polymerase. D, DNA helicase and DNA ligase. E, DNA polymerase and DNA ligase. Answer. Let me see. Come on, guys. We have 27 people here. I need more people to make this attempt. Or is the question difficult? Is it difficult? Okay, you guys are correct. DNA ligase is what opens it. You notice when I said it, it opens by DNA ligase. Sorry, DNA helicase. Sorry. DNA helicase opens it, right? Then DNA polymerase copies it, replicates it. That's what they're asking you. So the answer is DNA helicase and what? DNA polymerase. Some people are choosing ligase. No, ligase is what zips it back up. Helicase is what unzips. All right? Yes, and what they're asking you is unzipping. That's helicase. All right? Then photocopying. That's DNA polymerase. Okay, my understanding is cytokinesis finishes at antophase or anaphase. I think you wanted to say anaphase. Wouldn't it overlap there? No. So look, I want you to look at this picture. The person that asked the question. Don't worry, I understand what you're saying. Look at the picture. Can you see this picture? If you can see it, just type yes for me. If you can see it, type yes for me. Are you with me? Type yes, type yes, type yes. I'm not seeing any yes. Is my yes, okay. So if you can see this picture, look at it properly. It's prophase, metaphase, anaphase, and telophase. Did you notice this arrow? Look at this arrow here. The end of this arrow that is pointing inwards. Did you notice that this is cytokinesis? And why this arrow is covering both cytokinesis and telophase, not even anaphase, is because the two of them overlaps each other. Right? Remember, telophase is when they are going to the extreme. As they are going to the extreme, the class breaks into two. Breaking into two is cytokinesis. Going to the extreme is telophase. So the two of them actually overlaps. So when they break into two, the cell now rests. That resting is what? Interphase. But the place where the separation of the chromosome, remember, we're talking about the chromosome and the cell. Cytokinesis is breaking of the cell, not chromosome. So don't get it confused. When we are talking about breaking of the chromosome, it starts with anaphase. I agree. They come to the middle, metaphase. They start breaking apart, anaphase. All right. Then they now start going to the extreme. That's telophase. Before they get to that extreme, the cell breaks into two. That is cytokinesis. So cytokinesis and telophase, they overlap each other. All right. So that's the rationale. Very good that you asked. Very, very wonderful that you asked. Okay. Yeah, you guys are good. Okay, let me ask you this. No, this one is very, very straightforward. Okay, I will need you guys to give me the answer in less than 1 second. You're welcome. Feel free to ask any question, guys. Please feel free to ask any question. I'm here to take questions from you. Any question at all, to understand the concept. I'm here for you. Yes, IP math. I passed my anatomy test. C. I always put IP math C. I passed my anatomy test completely. All right. Okay, let's go. Meta. Everybody's metaing. Let's go. Yes, the answer is metaphase. You guys are good. Okay. Now, I'll give you this question. Look at this question, but you need to relax so that I will. Thank you, guys. It's metaphase. The answer is metaphase. Look at this question. I'll give you this question, but I'll put the option. So hold on. Let me put the option before you start answering so that you don't confuse others. This is A. Yeah. This is A. This is B. This is C. This is this is D. This is E. Okay, talk to me. So I've given you A to E. What's the answer? Okay, we already got one answer. Let's go there, guys. Let's go. 1 minute, 1 minute. I don't need you to take too much time on this. Your exam, it's fast and accurate. Huh? Which of these processes occurs during the cell's first gap? That's G1 phase. What happens in G1? Okay. I have D D D D D D D D D D D D D D D D. I should just go and release the music. Okay, the cell synthesizes protein. Yes, you guys are correct. You guys are very correct. The cell synthesizes proteins and what? Grows. Extremely correct. I agree. I agree. I'm not giving options for this. I'm not giving up some options for this. Let's see. Let's see who gets it. You guys are correct in the D, all right? You guys are very correct in the D. Sister chromatids are put to opposite poles of the cell during what? Exactly. Answer anaphase. Please answer anaphase. You guys are so smart in this class, man. I love it. Please, I hope you'll be like this during your exam so that you can, if they give you 50 questions, you can finish it in 20 minutes. Okay. So yeah. So let me give you this. Okay, try this. Try this, guys. Try this. Quickly. Try this. Chromatin shortens and thickens, coiling into compact rods during dash of mitosis. Answer. Let's go. Answer. Prophase. Yes, so shorten, thickens, all those things are preparation. P for P. This is prophase. You guys are correct. Very, very, very correct. I love it. Now, maybe we should start giving award for people, the first 10 people that answers question in this class. So maybe I will start doing that. All right. So let me try this question. Let me see the first 10 people that can attempt this question. But please hold on. I'll put options for the question before you answer. All right. Don't answer yet. Just think about it. Then I'll give you option. Just think about it. I'll give you option. This is, relax. This is option 1. A. This is A. Hold on. Please, I need you to think. Don't follow the crowd though, because I'm sure many people may fail this particular question. It's a little technical. This is B. Hold on. This is C. This is D. And this is, somebody's already writing P math. Okay, and this is C. And this is E. Okay, I need you to put either A, B, C, D, or E. Okay, you can say write it out if you have this the strength to write it out. Okay. Let's go there. Let me see. Let's go. We have 28 people here. Come on, guys. Let's go. Okay. D is missing interface. You guys are, let me see. Somebody said Did somebody say PB? Did he say B? Somebody say IP must C. Pass my anatomy test completely. Okay, you guys are very correct. You guys are extremely correct. Think about it for people that does not understand this, because I know that there are some people that didn't answer. So I take it that you don't really understand it. They say, which of the following omits a stage of the cell cycle? The first option says G1S, G2. That is interphase, correct? And mitosis. You know, we have interphase there, mitosis. So that one is complete. A is complete. B says interphase. Interphase is G1S and G2 and mitosis. That is complete. Then C says G1, DNA replication. What is DNA replication? DNA S. Then G2, then mitosis. It's complete. Then the last one, that's E, says interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. It's complete. But the D says prophase, metaphase, anaphase, telophase, cytokinesis. What of interphase? Interphase was missing. That's why D is the answer. So in the exam, I need you to think like this, all right? If you say a similar question, because you may not see these questions, but if you say, okay, let me ask, do you guys like the question? If anybody like the question, I want you to click the like button. There is a like button somewhere there. Click that like button if you like this particular type of question. Let me see. If you don't like it, put. Let me see. Nobody's liking this question. You guys don't like this question. The question is very okay. Somebody sent love. Okay, let's see. Let's see. Okay. 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 Good. Good. Okay. Good. Let me see 10 loves for that. Let's see. Let me see. Okay. 9. One more. One more. Come on. Okay, 10. Perfect. So that question is very interesting. Now, there is this question. I'm not giving you option, please. I'm not giving you option. This is a giveaway. I just want to do a giveaway now. Because of how you guys got that question, that's why I'm giving a giveaway right now. So this is the giveaway. Please don't be nervous in any exam you're writing. Tell yourself that you are beyond this exam. You are more than capable of this exam. You understand? Any exam you are writing, you should be going for 100 over 100. You can't waste. You can't spend all this time with me and you will not smash your exam. It has never happened in the history. It has never happened. Give me the answer to this question quickly, guys. I'm not giving you option. A karyotype shows the dash chromosome sorted and isolated from a cell in dash. 23 interphase, 23 metaphase, 46 prophase, 46 metaphase, 46 S phase. This one will be difficult for people, but let me see. Let me see somebody that can get this question right. Okay, for the first time, people are giving different answers. Somebody clean the answer because he's afraid. Please, the answer is. Please, the answer, I need you guys to go and study this thing later. The answer is 46 metaphase, all right? A karyotype, if you see that karyotypic chart, maybe I didn't mention it from the beginning of this class, it's 46 chromosomes, and also they are isolated from a cell in metaphase. It's always metaphase. Please just put that in your head, even if I didn't say it initially. So I understand why you guys didn't get it right. Many of you didn't, but it's fine. It's fine. I need you guys to, okay, somebody said I don't want mathematics, but mathematics keeps calling you. Mathematics keeps coming. It keeps coming. Tell me, what do you think about this question? What do you think about this question? Let's see. We have 30-something people here. So give me the answers. Come on, guys. Let's go. I don't want to give you options. I don't want to give you options for this question. I need at least 10 people to get this question correctly. A cell finishing mitosis has how many DNA molecules? Why a cell finishing DNA replication has how many DNA molecules? Come on, let me see. Somebody put. Oh God. Okay, let's see. Somebody say 46, 92, 46, 92, 46, 92. Let's see. Let's see. Okay. Okay. Come on. I need to get up to 10 people here. Come on, let's go. Come on, let's go. Come on, let's go. Option 46, 46, 46, 92, 46, 23, 23, 46, 23, 23. Anybody want to change their option? The answer is 4692. The answer is 4692. It's just this is not even mathematics. This is just common sense. They say a cell finishing mitosis. It just finished mitosis. So it has to be 46. It's normal. But when it has replicated, what does replication mean? Dividing into 2. That 46 will become what? 92. That's photocopying, like photocopying itself. So the 46 becomes 92. It's straightforward. For real. Though somebody said they don't like this question, so that's fine. Maybe something like this may not come out, but it's actually very simple. It's actually extremely simple. All right. I'm coming to the last among the last questions in genetics because I need to jump over to tissues. I need to jump to tissues. Tell me this. Answer very quick, no time. I'm not giving options, please. Wow, nobody's talking. Okay, please let me give options though. I'll give you options. A, pleiotropy. B, sex linkage. C, polygenic inheritance. D, incomplete dominance. E, multiple carriers. Answer. Come on, let's go, guys. Let's go. Hit it up. The more you attempt, the better. I need you to reason it out. Eye color is caused by genes at multiple loci. You remember what I said? Something about multiple loci, causing one particular thing, like my skin color, like eye color. Perfect. You guys are extremely correct. It is polygenic what? Inheritance. Right? And the opposite, the other one is what? Pleiotropy. Right? This is polygenic inheritance. This is very, very correct. Very correct. Thank you, guys. Now, for that, let me give you this bonus question. Okay. Talk to me about this question. Germ cells have dash unpaired chromosomes and are thus called dash cells. 23 somatic, 23 haploid, 23 diploid, 46 somatic, 46 diploid. Answer. No, the option again is 23 somatic, 23 haploid, 23 diploid, 46 somatic, and 46 diploid. Let's go. Exactly. You guys are wonderful. It's 23 haploid. All right. Let's see this. 23 haploid. So give me the answer to this one very fast. Option: species DNA, population DNA, gene pool, genetic drift, phenotype. Species DNA, population DNA, gene pool, genetic drift, phenotype. Sorry, I have to be a little faster, right? Please. Pardon me. Let's go. Exactly, Jimpu. Jimpu, you guys are correct. So let's go in this one. I'm being very fast right now. Let's go. Let's go. Let's go. Do you need options for that? Okay, fine. I'll give you options. Germ cells, haploid, A. Germ cells, diploid, B. Somatic cells, haploid, C. Somatic cells, diploid, D. Answer. Come on, guys. Speed it up. Speed it up. Exactly. You guys are correct. It is what? Somatic what? Diploid. Thank you so much. Now, let me see. I don't know if this diagram will paste. Trying to paste the diagram and it's not pasting. It's not. It's not pasting. Unfortunately, it's not pasting. Okay, fine. So, let me see. Yes, you're correct. You're correct. Oh, somebody is still in gene pools. We are way beyond that. Okay. So, anybody has a question? This is the much you can see in genetics. Well, any questions? It has to be something similar. You understand? Then look at your diagrams properly, and you'll be fine, good to go. Anybody have a question? No question. I'm jumping to tissue very fast because we've really spent almost an hour 23 on this. And tissue is something very important also that you need to know. All right. Now, please, I need you to just give me 10 minutes before I jump. Okay, give me 15 minutes. Let me discuss this issue before I jump into the questions immediately. Don't sleep on me, please. Don't sleep on me. I know you don't need a break. I know you don't need a break because this is crash course. We are doing crash course. All right. Tissues is very important. It's important for you to know the categories of tissues, which are the epithelial tissues, the connective tissues, the nervous tissues, and the muscular tissues. 4 of them. ECNM, epithelial tissues, connective tissues, nervous tissues, and what? Muscular tissues. All right? Very, very important. Now, what do you need to know in tissues? You need to know about the types of tissues, the characteristics of the matrix, and relative amount of space occupied by this matrix. Please, if I'm speaking very fast, I'm sorry. Later, you can actually watch this. It will be on my YouTube channel. All right. I know all of you are subscribed already, so you can just go to the live section and you will see this video again. All right. So it's not a problem before your exam tomorrow. For people that are writing tomorrow or whatever, for your professor. All right. Tissue, you need to know what tissue is. Tissues are cells, similar cells that are grouped together. All right. That's something you need to know. I need to know what matrix is. Matrix is just more like the surrounding. All right. Surrounding. It can be fibrous proteins. We also have clear gels called ground substance, also known as tissue fluid. So we have tissue fluid, we have ECF, and we have interstitial fluid. Tissue fluid, ECF, interstitial fluid. Extremely important for you to know. We'll still look at it very well. This is one question that they always ask, which is the primary germ layers: ectoderm, endoderm, mesoderm. I'm not going to spend time here, but ectoderm is always outside. It gives rise to what? Epidermis and nervous system. Skin. And if you can touch me, you feel something. That is ectoderm. Simple. Endoderm is the innermost. All right. Inside. It gives rise to what? Things like digestive system. Like if I open my mouth now, you go down, that's digestive system. So it makes sense. So if you see digestive system, respiratory system, you are thinking of what? Endoderm. Inside. Now, in between endoderm and ectoderm, in the middle layer is known as what? Mesoderm. What do you see in mesoderm? You see bone and blood. Check it out. You understand? So if you know it like this, you are good to know. And you need to also know histological stains, right? Like the smear, like the spread, all right? And also the sectioning. You need to know the difference between longitudinal sections, cross sections, oblique section. All right? You need to know these things because they can ask you a question. Oh, a tissue cuts on its long axis. That's longitudinal section. All right. So you need to know these very, very well. You need to know this the functions of epithelium. I believe all of you knows that. You need to know what basement membrane is. Like layer between an epithelium and an underlying connective tissue. The basal surface, the apical surface. Now, this is extremely important. You see these shapes? They are extremely important. They are extremely important that you know this because I am very sure that any exam that you do on cells, on tissues, especially your lab exam, you have to see this. You have to know how to identify, all right? You can identify in two ways. One, by the amount of layers that's standing on each other. Anyone that has layer on each other, that's stratified. Anyone standing alone is simple. Then for the shapes, you have the one that looks like a pizza, which is the squamous, the cup, the cuboidal, the columnar, the tall ones. All right? You have to know this. If you look at this diagram, if they ask you about this diagram, you're talking about stratified. You need to know where is stratified epithelium. Where is it found? All right? That particular stratified, where is it found? Now, we have the four types that are in one layer, which is the simple squamous, the simple cuboidal, the simple columnar, and the pseudo-stratified columnar. Now, they can describe it for you. They may not tell you that this thing is simple columnar. They may tell you, oh, tall, narrow, tall, narrow. Just, you know that it's simple columnar. Round or square, you know that it's cuboidal. Thin or flat or scaly, you know that it's simple squamous. Then we have pseudo-stratified. That's the one that looks like it is simple columnar, but it's stratified columnar, but it's not. Columnar, you can never see stratified columnar. Just remember that. So anyone that looks like stratified columnar is what? Pseudo-stratified, because it's trying to confuse you. Don't get confused. And this pseudo-stratified, they always have goblet what? Cells. Goblet cells produces what? Mucus. That's what you need to know. All right? Now, what you need to know is where can you find each and every one of them? Where will they be testing you on? Where will they be testing you on? Start with simple squamous. Simple squamous, where are you seeing it? You are seeing it because where places that secrete serous fluid, specifically serous, not mucus. Now, where are we talking about? Usually alveoli. That's one place that they will test it. It's also found in the glomeruli, actually. Specifically in the nephron, the particular part of the nephron, the thin descending loop of the nephron is simple squamous. But the place they usually test for simple squamous is what? Alveoli. Put it, close your eyes right now. Put it in your head. Alveoli is simple squamous. Alveoli is simple squamous. Alveoli is simple squamous. I'm too dramatic, sorry. Okay, now look at this place, right? Cuboidal. I told you cuboidal is it's like just like a cup. Anywhere that produces fluid, like the liver, it produces fluid, thyroid, mammary gland, all the glands. Because glands produces what? Fluid. Bronchioles, kidney tubules, all of them are what? All of them are what? Simple cuboidal. Simple cuboidal. But whenever you see stratified cuboidal, anytime you see stratified is talking about some places that it need to be hardened. It needs to be hard. So stratified cuboidal, for example, is tracts where the fluid that is produced by the glands passes through. So, for example, if you see mammary gland now, you are talking about simple cuboidal. But if you see ducts, you are talking about what? Stratified cuboidal. That's the wisdom. Now, you have simple columnar. That one, you usually see it in the GI, all right? GI tracts. You can also see it in the uterus and some parts of the kidney and uterine tubes. So, two places I need you to know for the simple columnar is what? GI and the uterus. That's the two places you need to know, most importantly. Pseudo-stratified is usually found in the trachea. That's the respiratory tract. You can never forget this one. All right? Now, when you are talking about the stratified columnar, what do you need to know? Do you remember what I told you about, the stratified epithelium? Do you remember what I told you about the stratified columnar? It is rare, extremely rare. All right? So we are not even talking about it. So what do we have? We have stratified squamous and stratified what? Cuboidal. Those are the two you should pay attention to. Also, we have the transitional epithelium. Transitional epithelium is also important. Where do you see transitional epithelium? Ureter and bladder. Chicken. Let it sink. Let it enter your head right now. All right? So because, this is crash course. Now, when you are talking about stratified squamous, you are seeing it in the only place that stratified squamous and keratinized is usually the skin. Also, we have the hard palate. This part, the hard palate, is keratinized. But if they ask you about keratinization, please look for the skin. That's where you hardly see keratinization inside the body apart from the hard palate. That's all. All right. So non-keratinized are usually the other surfaces. All right. But keratinization, you need to know. So here we have keratinized stratified squamous. Where do you see it? Skin. Everything is skin. All right. Keratinized squamous. Why non-keratinized stratified? You see it in the vagina because usually the vagina is a cuboidal. You understand? But it's changed. That's why we call it metaplasia. It changes to what? Non-keratinized stratified what? Squamous. Extremely important for you to know this. Extremely important. Because they ask the vagina a lot, they ask the tongue, they ask the esophagus for non-keratinized stratified squamous. So you need to know, where do I find keratinized? Skin. Where do I find non-keratinized? Vagina, esophagus, oral mucosa, tongue, even the anus. Stratified cuboidal, where do I see it? Ducts. You see what I was telling you? Sweat gland ducts. Ducts. You can also see it in seminiferous tubule. Okay. Ovarian follicles. Okay. Ducts. It's stratified cuboidal. Please don't forget. Transitional epithelium, ureter, and what? Bladder. And that is it. That is it for tissue for epithelium. That's one part of tissues. Another part is connective tissue. Remember, I told you connective tissue is always under the epithelium. Epithelium is usually what? Avascular. Connective tissue is under, it gives it what? Blood. All right. So let's look at connective tissue. It's very, very, very vascularized, extremely vascularized, supports organs, connects and protects everything. These are the functions. Connecting organs supports physical detection, immune protection, movement, storage, heat production, and transportation. These are the cells: fibroblasts, macrophages, leukocytes, plasma, mast cells, adipocytes. These are the fibers: collagenous fiber, reticular fiber, and what? Elastic fiber. They may ask you questions on this fiber. It's important that you know some of the examples of this fiber. All right. When you are talking about collagenous fiber, you are usually talking about tendons, you're talking about ligaments. Reticular fiber, what are you talking about? You are talking about what? Spleen and lymph nodes. Elastic fiber, where do you find elastic fiber the most? It's usually a protein called elastin, right? That allows it for stretch. I will show you the place you see it a lot, all right? Before that, you talk about ground substances, things that are found within the layers of the connective tissue. We have what? GAGs, that's glycosaminoglycans. Extremely important that you know it. Now, what are they going to ask you on glycosaminoglycans? They will ask you, one of the things is a gelatinous, one of the gelatinous materials that are found in the connective tissue. Something like that. I would look for that question. Similar questions to it. So what do you see? Glycosaminoglycans. What makes up glycosaminoglycans? First of all, you should know that glycosaminoglycans, that's GAGs, they regulate what? Water and what? Electrolyte balance. Water, electrolyte balance of the tissues. You cannot forget this. Water, electrolyte balance of the tissues. I'll give you a question. You will see what I'm talking about. Talk about chondroitin sulfate. Talk about heparin. Talk about hyaluronic acid. If you see this is an option, you should be able to know, okay, they are talking about the gelatinous substances, that the glycosaminoglycans. Also, they talk about proteoglycans. All right. They talk about other adhesive proteins. I'll give you questions. So I need you to remember. And remember, when I classified connective tissues, I said that we have connective tissue proper and we have what? Specialized what? Connective tissue. Sorry. Connective tissue proper and specialized connective tissue. Then in connective tissue proper, we have the loose, we have the dense. In the loose, we have the areola, we have the reticular, and we have the adipose. In the dense, we have the dense regular, dense irregular. I hope you can remember, all right, for people that have been able to teach in my class, all right? So I need you to remember, this is areola. This is areola. It's under the epithelium. Very, very important that you know it's under the epithelium. In serous membrane between muscles, passageways for nerves and blood. Reticular is found in the spleen and thymus. If you notice what we discussed here, this is reticular fibers. Where did I say we find reticular fibers? Spleen, lymph. So you need to remember that. Extremely important. Then we have the dense regular, all right? Dense regular. Tendons. Tendons are major examples of dense regular. Tendons. We have the dense irregular. Dense irregular. You understand? Deeper layers of the skin, capsules of that are around the organs. That's where you usually found it. Then we have the adipose tissue. White and brown fat, fine. But you need to see the adipose tissue is empty looking. Extremely important that you know that it's empty looking. All right? That's beside the point. Let me give you a formula for chondroblast and chondrocytes. Anytime you see anything, they ask you a question about lacuna, please go and choose the sites. Any question, and they're asking you about maybe osteocytes, look for lacuna. Because the sites, they are usually cartilaginous cells that are what trapped in where lacuna. Anytime you hear sites, both chondrocytes or what osteocytes. Why the chondroblasts are cartilage cells that produce the matrix that traps them. All right, extremely important that you know this as well. So this is the cartilage. This is the cartilage. All right. This is cartilage, like hyaline cartilage, where it's located, trachea and articular surfaces. This is elastic cartilage in the ear. Please remember this one. This one is very important. Elastic cartilage is, you know, your ear, external ear, also the epiglottis. Then this is fibrocartilage in the meniscus and also the intervertebral disc at your back. All right, that's in the vertebra. Now, in terms of bone, you understand, we have two forms. We have the spongy bone and we have the compact bone. Spongy bone and compact bone. But one of the most important things in bone is this diagram, the structure. All right, the structure is also very important because it looks like onion. Look at it, it looks like onions. So if you see the bone, you will know. And extremely important is the cells. We have the osteon, that's the central canal. Then we have the osteocytes. What did I say about sites? Sites within what? Lacuna. Within lacuna. See how I'm putting my ears as if I can hear you guys. All right. So that's what you need to know. And also, specialized connective tissue, we talk about the blood and the bone, right? We've discussed the bone. This is the blood. These are the cells of the blood. Extremely important that you know. All right. Now, in terms of excitability, nervous tissues are very excitable. They have the ability to respond to stimuli by changing membrane potential. Same thing with muscular tissues. All right. And membrane potential is the electrical difference. All right. So you have to bear this in mind. And for nervous tissue, you need to know that the nervous tissue has a specialized communication through electrical activity and chemical signals. All right. They detect stimuli and respond to it very fast. And they have something we call the neuroglia. That's the glia cells. These glia cells are what protects and assists the neurons. They wrap up the neuron. They protect it. All right. So when you are talking about nervous tissue, you are talking about the neurons and what? The neuroglia. Remember that. You will see a question on that. So these are the parts of the nervous tissue. We have the body, that's the neurosoma, where the cell is housed. This is the dendrites, and this is the axon. Extremely important that you know these three things. Now, when you talk about the muscular tissue, what you need to concern yourself most about is the types of the muscular tissue to be able to differentiate them. We have the skeletal. I know I'm speaking very fast. I'm so sorry. I'm trying to, be able to micromanage my time because we've not even taken questions. And trust me, I know how to push a lot of questions. And one thing you need to know is that the more questions you answer, the better for you. Trust me. You have this. Once you get one question, get the second one. You'll be like, oh, this is very simple. Something that is very difficult for you will suddenly become simple for you. So my revision classes, I'll be revising for you guys now very often. My revision classes like this, we have to treat a lot of questions, all right? Now, important source of heat, body heat, we have the skeletal, we have the cardiac, we have the smooth, all right? Skeletal, cardiac, smooth. These are the important muscular tissues. They are very, very important for body heat, all right? When you are cold now, you shiver. Your muscles shake and vibrate for you to be able to generate what? Heat, all right? I hope you guys are following me. If you are following me, please click the like button. Just click the like button so that I know that people are still here. We have 26 people here. I need to see 26 likes, all right? So as you are doing that, I'll continue. We have the skeletal muscle. What I need you to know is the differences in terms of striations and in terms of voluntary or involuntary. Skeletal muscles they have striations, all right? And those striations are caused by alternating dark and light bands. Alternating dark and light bands. And skeletal muscles are voluntary. That is what you need to know. That is what you need to know. It's as simple as that. If you know that, you are good to go. Because when we will discuss muscular system, we will discuss this specially. All right? Now, we have cardiac. Cardiac are striated, but they are involuntary. They are striated, and what causes their striation is what? The intercalated disc. This is something that they will ask you in your exam. What causes the striation? Intercalated what? Disc. All right? Very, very important. And they are involuntary. Now, smooth muscle, if you look at this, the smooth muscle, you see, they are like fishes that are swimming. Very, very smooth. Very smooth and nice. All right? So they lack striations completely, and they're what? Involuntary. As a recap, skeletal muscles, they have striations by alternating dark and light bands, and they're what? Voluntary. Cardiac muscles, they are striated by intercalated disc, and they are involuntary. Smooth muscles are not striated, and they are involuntary, and they are usually found around what? Organs. Extremely important. All right? Okay. So I think this is the final part. Cell junctions. You need to know cell junctions. We have the tight junctions and we have the gap junctions. So tight junctions is when they are together. Tight junction, two cells being tight, nothing passes through. You understand? Then suddenly they have gap junctions. Things can pass through. Then they have desmosomes. Desmosomes are like patches that hold them together. Desmosomes, patches that hold them together. Something like this. You understand? Patches that hold them together. All right? Why hemidesmosomes are where they stand on. If you remember this, you are good to go. I'll give you questions, all right, so that you can cement it in your head. I'll give you questions. These are diagrams, very, very important for you to know. Tight junctions, everything is tight, closely held together. These are desmosomes. They are the things that helps to link them together. These are gap junctions, and these are hemidesmosomes. This is hemidesmosome around the basement membrane. Okay. Now we have glands as well. Glands is something that definitely will come out in your exam. All right. I hope your professor reviewed this. Glands are very important. We have exocrine glands and we have endocrine glands. Listen, what you need to know is that when you are talking about exo and endo, we are always talking about their conduction. Endo and exo is talking about conduction. When you are talking about exocrine, we are talking about two cells that are close to each other. How do they connect each other? Through ducts. Because they are close. They connect themselves through ducts. But endocrine glands are far from each other. So how do they connect themselves? Through blood. So when you're talking about exocrine and endocrine, you're just talking about how do they communicate. Chicken. One of the questions that they'll be asking you is they'll tell you, oh, sweat glands or tear glands is example of what? Think about it. Tears. Does tears enter your blood? No. That means it must be exocrine. I was discussing with someone one time, and the person was telling me, no, pancreas is endocrine. I say yes, because it produces insulin and glucagon. Yes, I agree. Endocrine. But pancreas serves two functions. Pancreas has an endocrine function and what? An exocrine function. What was the exocrine function of pancreas? Remember, pancreas produces what? Enzymes, digestive enzymes. The question is, how does those digestive enzymes move? Do they enter the blood? No. Instead, they move through ducts. That's why you have what? The duct that connects the pancreas and the duodenum. You see what? Pancreatico-duodenal what? Duct. Do you understand? So that's what you need to know. Examples of endocrine glands, things that moves in the endocrine. We talk about hormones, right? So anything hormone, endocrine, all right? Remember that. Now, another thing you need to know is let me see. This is important, but I don't know if they will ask you that in exam. Okay, so we have types of secretion. You should also know types of secretion. We have the serous, mucous, and mixed. Anything serous is water, like sweat, like milk, like tears, like digestive juices. That's serous. But mucus are the things that are, like mucin, you understand, glycoproteins, all right? Then we have the mix. Now, another place I need you to lay emphasis on is the merocrine modes of secretion, merocrine, apocrine, and holocrine. Merocrine, apocrine, and holocrine. They will ask you, tear glands, pancreas, digestive glands are examples of what? Merocrine. They are used by acrine glands. They are used by what? Acrine. So merocrine and acrine, the same thing. They use vesicles. They release their substances through exocytosis. When you talk about apo is anything that has what? Lipid. Simple. Like what? Milk. Chicken. And when you talk about holocrine are the ones that cells that accumulate a product until what? They disintegrate. They commit suicide. Right? Like oil glands of the scalp and skin. Listen, what you need to understand is examples of each. Tears, pancreas, digestive juices, all of them are what? Merocrine or acrine. Merocrine or acrine. Apo is anything lipid, like what? Milk. Holocrine secretion is anything that is like the scalp of the head. Scalp, like the oil that come out in the scalp, and also oil in the skin. They are holocrine. So merocrine, apocrine, and holocrine. I'm laying emphasis on this. For people that have ears, let them hear. All right. So we also have membranes here. A lot of membranes that covers up the body. We'll discuss this in tissues. Now, finally, for the tissues, you need to know hyperplasia, hypertrophy, neoplasia, differentiation, metaplasia. You need to know these things. I don't want to stress too much on this. Hyperplasia is just multiplication. I get a wife, I give birth to a lot of children in this room. That is what? Hyperplasia. We cover this room through multiplication. Or I decide to, okay, let me be myself. Let me just eat a lot and swell and cover this class. I become what? Hypertrophy. Neoplasia is anything that has to do with cancer. So if you just see cancer, please don't think too much. Don't think too deep. People fail questions because they think a lot. Too much thinking doesn't help. So that you don't forget what you know. When you're talking about differentiation, like most of you are nurses already, all right? Now, when you want to specialize into mental health, this one, that one, gynecology, pediatrics, you understand? That's what we call specialization. That's differentiation. So differentiation is specialization. Undifferentiated cells are called what? Stem cells. They are undifferentiated. Now we have metaplasia. Metaplasia is changing from one epithelium to another. Like smokers, usually the epithelium of the lungs, I told you, the trachea, the respiratory airway is usually what? Pseudostratified epithelium. When it changes because of cigarettes to become what? Stratified squamous, right? That's metaplasia. When the vagina changes from cuboidal, you understand? Cuboidal to stratified squamous, it becomes what? Metaplasia. So that's as examples of metaplasia you need to know. Then we have stem cells. I mentioned it is undifferentiated, all right? That undifferentiated stem cells can be divided into embryonic and adult stem cells. The embryonic stem cells, we have the totipotent and pluripotent, all right? While the adult stem cells, we have the multipotent and unipotent. So MU, this is PT. PT is PT, pediatrics, embryonic, PT, pediatrics, pluripotent and totipotent. That's for pediatrics. I'm just saying pediatrics based on embryonic. Then adult is MU, multipotent and unipotent, maturity. All right. So please remember that. Just knowing the examples is what will help you. Then we have regeneration. That's replacement of dead or damaged cells by the same types of cells as before. All right. When you injure yourself, it regenerates and you are fine. But there's something we call replacement by damaged cells with scar tissue. Instead of it to be replaced by exactly the same cell, it's now replaced by what? Scar tissue. That is called what? Fibrosis. Who is sleeping? Wake up. I'm talking as if I can see anybody. Please don't sleep on me. Don't sleep on me. All right. This is wound healing. Wound healing is very important. You understand? Healing of a cut in the skin, several vessels bleeding to, blah, blah, blah. We have the mast cells, that try to release histamine. Histamine dilates blood vessels and makes capillaries more what? Permeable, so that, blood can move to that particular site of injury. Now, what does the blood do? The blood carries antibodies and what? Clotting factors to clot. Yes, keep liking, please. Keep liking. I need this like to get to up to 30. You guys are up to 30 here. All right? To show that you are following me. Okay, so I will jump this for now. We'll jump this. Wound healing, you can read it up later. Tissue engineering, read it up later. Now, I want to talk about atrophy. Atrophy is the shrinking of a tissue through loss in cell size or number. Opposite of hypertrophy and hyperplasia is what? Atrophy. Remember, hyperplasia is what? Increase in size by multiplication, right? Increase in size by the multiplication of the numbers. Why? And hypertrophy is increase in size by swelling. All right. The two of them, opposite of them is what? Atrophy, atrophy, or whatever. Atrophy or atrophy. That's shrinking or a tissue through loss in cell size or number. All right. Now we have necrosis. Necrosis does it sound normal? It sounds pathologic, right? Yes, it's actually pathologic. It's pathology. Pathological tissue death due to what? Trauma, toxins, or infection. Tissue will start dying. Example of necrosis, we have infarction, we have gangrene. Under gangrene, you talk about ulcers like decubitus ulcer, dry gangrene, wet gangrene, and gas gangrene. All right? Yeah. Very, very important you know this. Then we have apoptosis. Apoptosis is programmed cell death. Listen, you need to just get these terminologies in your head. Like atrophy, like we have hypertrophy, hyperplasia, hyperplasia, metaplasia, neoplasia, differentiation. 5 of them. Put them in your head. Now, here we have atrophy, that's 6. Necrosis, that's 7. Apoptosis, programmed cell death, that's 8. You should be able to differentiate all of them. Because if I give you a question, you will see what I'm talking about. Okay, that's the last part of the slide. Eureka. Who is ready for question? I hope you guys are not tired of me. Oh, please, don't be tired of me. I really need everybody to pass here. Everybody to do well in the exams, right? So let's go. I hope you guys are ready. I hope you are ready. So let me start with the simplest question so that I don't discourage people. I don't make you run away. Are we ready? So I'm going to put this question with the option in the box. And no, I'll just put only the question, then I'll give you the options, then you write A or B or C. Please don't sleep on me. If you are sleeping, wake up yourself. Wake up yourself. Don't sleep. All right? This is just 10:52. We still have an hour to go. All right? Yeah. So, but this time around, I'll be very fast. Okay. Let me see who is awake. Okay. We are 33 people here. I need at least 20 people to answer before we move. The options are A, connective tissue, connective, B, fibrous, C, nervous, D, epithelial, E, muscular. The 4 primary tissues found in adult organs include all of the following except dash tissue. A, connective, B, fibrous, C, nervous, D, epithelial, E, muscular. Oh, wow. People are still awake. Come on, let's go there. Let's go there. Come on, let's go there, guys. Let's go there, guys. I still have 1, 2, 3, 4, 5, 6, 7. No, I need up to 20 people to make attempts before. Up to 20 people to make attempts before I Let's go there. Let's go there. Let's go. Answering. A, connective. B, fibrous. C, C is what? Nervous. Then we have epithelial and we have muscular. Okay. Okay. Yes. The answer is fibrous. Fibrous is not parts. Eh? This is a giveaway. Oh. Don't think this one is a big question. I'll give you the main question. This one is just to wake up everybody that is sleeping. To wake up everybody that is sleeping. Okay. Hold on. Let me archive out the serious questions. This one is just an unserious question for now. Okay. So I'm happy that all of you are still awake. I'm so sorry. This one I'm not giving you an option. Not giving you an option here in this one. So answer. These ones are just to wake you up. These are questions to wake you up. I need everybody awake. Everybody should be blazing right now, answering these questions. What is the answer? The gel inside the cell is called what? I need a lot of, just put anything. Put anything. Come on, guys. Come on, guys. Let's go. Come on, guys. Come on, guys. Let's go. Come on, guys. Let's go. Blaze it. Blaze it. Let it get hot. Come on, guys. Who is there? Come on, guys. Come on, guys. Okay, fine. Some people answered cytoplasm, some people answered cytosol. The answer is cytosol, all right? Another name for cytosol is intracellular fluid, correct? Not cytoplasm. Cytoplasm is the space inside. Do you understand? Space inside. Why cytosol is the gel? So that's why it was very specific, all right? Now, I want to give you this one, and I need you guys to hold on. I need you guys, yes, cytosol. I need you guys to hold on. Let me paste the answers before you pick, all right? So this is the question. This is the question. Then this is the option. Wow. I thought we were up to 30. What happened? Are we now 20? They ran away. Come on, guys. You don't run away right now. This is A. Hold on. This is A. Am I being too slow? Sorry. I hope I don't burn you out. This is B. This is C. This is D. And this is E. Come on, guys. Let me see people picking the answers ASAP. All right? Come on, come on, come on, come on. Everybody has to make attempts. Everybody. Okay. People are shooting. Come on, guys. Come on, guys. Let me see. I have like 20 people already making attempts. Where are the remaining 8 people? You are up to 28, 30 here. Come on. Everybody must pick an answer. It's fine if you are wrong, all right? Okay, that is correct. You are absolutely correct. Ectoderm, the ectoderm gives rise to what? Nervous system and what? Epidermis. That is good. That is good. I don't want to start reviewing the other ones, because I don't have a lot of time for it right now. So, sorry. This one, I will give you the option. I will call out the option. So I need you to make attempts. Everybody. Option. Let me see the fastest people here. Everybody. Option A, fixative. B, histological section. C, spread. D, stain. E, smear. Fixative, histological section, spread, stain, and smear. Come on, everybody! I need everybody to make attempts. I need you guys to make attempts. Okay, perfect. Perfect. You guys are very engaged. You guys are good. This thing was 35 before. Now it's 19 people. Where are they running to? They don't need to run, no, because we've not even gotten to the main questions for tissues. Better hang around, guys. Hang around. Okay, so we have 18. Okay, so the answer is histological section. Very important. Now look at this question. I'll give you options from my end here. Okay, we have options is cross, oblique, transverse, sagittal, longitudinal. Cross, oblique, transverse, sagittal, longitudinal. The histological section that divides a specimen along its axis is called dash. I need more than 10 people to make attempts on this question. Come on, guys. Come on. Let's go. Cross, oblique, transverse, sagittal, and longitudinal. Let's go. We have 23 people there. I need 20 people to at least answer this question. Come on. We have 25 people right now. Let's go. Keep moving. Where are the remaining 10 people? We are supposed to be 35. Where is everybody? You guys are sleepy already. Please don't sleep on me. We have less than an hour to go. Or more. Depends on when we finish anyway. Yes, the answer is longitudinal section. The answer is what? Longitudinal section. And I told you that question is very important for you to know. That question is extremely important for you to know. All right? Yeah, because if you remember, let me see if I can remember exactly where I found that question. If you look at this particular slide, yes, they can ask you a question from any of these, right? Yeah, the answer is longitudinal. Thank you. They can ask you a question from any of these. Longitudinal section are tissues cut on its long axis. This is long axis. You see how it's like this? You cut it. All right? That's longitudinal. We have the cross section. Tissue cut perpendicular to the long axis of the organ. You need to listen to the wording so that you'll be able to get the answer correctly. This is perpendicular. Then we have the oblique, tissue cut at an angle between cross and longitudinal section. All right? Please, this is extremely important. You understand? So you need to, because I just gave you one of the ways they can ask questions. They can ask you either the cross or the transverse. You never can tell. All right? So I need you guys to be able to understand that. Okay, I need you to attempt these questions for me. Let's see, fastest finger. The basement membrane is found between dash and dash. You want options? Fine, I'll give you options. A is epithelium, connective tissue. B, epithelium, extracellular material. C, epithelium, intracellular material. D, extracellular material, intracellular material. E, interstitial fluid and extracellular fluid. Come on, guys. Come on, guys. Let me see. Hit that. Hit it. Let's go there. Where is it? 10 people. You people are supposed to be 35. There are like 10 more people that was here before. They disappeared. So let's go. The basement membrane is found between dash and dash. Let's go. Epithelium, connective tissue, epithelium, extracellular material, epithelium, intracellular material, extracellular material, intracellular material, interstitial fluid, extracellular fluid. Come on, guys. Don't disappoint me. Some people are shying away from picking answer. I need everybody to, okay, good. We are around back to 33 people. I need everybody to pick an answer for me. You can be wrong, it's fine, but this is just a rapid review. Come on. Okay, good. The answer is what? A, epithelium and what? Connective tissue. Remember I told you that the epithelium is here, standing on the basement membrane. This is the apical part of it. The up part, this is the apical part up here. This is the basement membrane, the side facing the connective tissue. And I told you that the epithelium is what? Avascular. And because it's avascular, it needs something to give it what? Blood. It needs something. It's a vampire. It needs something to take blood from. And what does it take blood from? From the connective tissue. So the epithelium, the basement membrane is just the place where the epithelium is putting its leg on to trap blood from the connective tissue. You need to remember this. And which type of connective tissue is usually found below the epithelium? The loose connective tissue. Majorly the areola. You cannot forget this. Yes. Answers are still coming in. Nice. You know, we are still testing the waters. We've not even gone deeper, right? So we are going to go deep gradually. Let me see. Good. I'm not giving options here. No, I'm not giving you options here. I will not do that because you are supposed to impress me. I see that there are some people that are not in the United States here. That's good. That is good. If you've not subscribed to this channel, please subscribe because we'll be having a lot of these sessions. Make sure that everybody passes their exam. If you have any topic you want to discuss anytime, always feel free to send me a message here on, just make a comment, all right? Extremely important. Watch the videos. Okay, let's see. Wow, these people are smart. You guys are hot. Fire is coming out from your head. All of you. Okay, good. We are back to 36. Perfect. So meaning that the people that were sleeping before, they've woken up. Perfect. I love that. So rapid diffusion of oxygen and carbon dioxide through the S sacs, that's the alveoli of the lungs, takes place through dash epithelium. Answer. Come on. More people. Put it in the box. Perfect. Simple squamous epithelium. I told you the places you find simple squamous epithelium in the body is where? The alveoli. You can also see it in the kidney, but which part of the kidney? Remember, the kidney is usually the cuboidal, right? So which part of the kidney can you find simple squamous? It's usually the thin descending loop. If you do urinary system, if you've not done urinary system, please go and watch my urinary video. You will understand urinary a lot. All right. You see that there is a place where water goes out from. You understand? Something we call we call it reabsorption. All right, because we have filtration, reabsorption, then secretion, and water conservation. So water conservation, reabsorption usually happens in those places. All right. So I need you to remember that. I'm not doing urinary now, so I'm not discussing urinary. So the answer is what? Simple squamous. If you look at that option, cuboidal, where do you see simple cuboidal? Mammary gland, liver, thyroid, places that produces what? Fluid. See how I put that D at the ending so that you don't forget. Simple columnar, where do you see it? The digestive system, like the stomach, like the intestine. Chicken, no stress. All right. Then we have the keratinized stratified squamous epithelium. Where do we see keratinized stratified squamous epithelium? Please, I need you to put the answer for me in the comment section. I need 10 people to give me the right answer. Where can we find keratinized stratified squamous epithelium? There is no exam that you do in tissues that you will not see this question. Be it NCLEX, be it anything, you must see this question in it. Where? Where? Come on, keep going. I have 4 people. Keep going. I need 10 people to tell me the answer. Okay, keep going. Keep going. Keep going. Let's have it. Let's have it. Keep going. Keep going. Keep going. Perfect. Okay, I have 10 people. You guys are wonderful. So you have it. So keratinized is always in the skin. Skin. For your exam purposes, please remember the skin. And for people that are writing NCLEX, please, when you are picking the skin, also remember the hard palate. The hard palate. Those are the 2 places majorly you find stratified, keratinized. That keratinized is very important. But non-keratinized, you find it where? The tongue, the esophagus. You can also find it in the vagina and the anus. That's the non-keratinized stratified squamous. I am trying to push this because it's something that comes out a whole lot. Thank you. Okay, let me see. Let me try you with this. Guys, let me try you with this. Please, I will read the options. So pay more than usual attention so that you'll be able to get. Pay more than usual attention so that you'll be able to get the option. Are they sleeping? Why is the number reducing from 35 to 18? Please don't sleep. Call them to come back. Or they are having a problem logging in. So give me the answer. The option is A. Okay. Since people have started answering, I'm not giving option anymore. Let me see the people that are following me all along. Oh, it stopped. Please let me give you option before you choke me. A, simple columnar. B, stratified columnar. C, pseudostratified columnar. D, simple cuboidal. And E, stratified cuboidal. Answer. Keep going. Keep going. Keep going. Keep going. Let's have it. Let's have it. Keep going. Wow. You guys are so brilliant, honestly. Like, I am sure that if you are preparing for NCLEX here, you are good to go. The only thing you need now is just to clinical application. For NCLEX purposes, clinical application. But right now, you guys are already ready. Yes, the answer is simple cuboidal. You are extremely correct. If you see that, I told you, cuboidal is usually found where it's producing fluid. Cuboidal. Cup. Where produces fluid. But if it is stratified cuboidal, it's usually tracts or seminiferous tubule, where things pass through, not where it produces it. I need you guys to remember this. Okay, I put another one there. Give me the answer. Okay, no, I'll give you options. This one, you need options, right? You need options. That epithelium provides a moist and slippery surface and is well suited to resist stress, as seen in the dash. A, please pay attention for the options. I'll read it only once. A, stratified tongue. B, keratinized tongue. C, non-keratinized skin. E, keratinized skin. D, keratinized skin. Then E, non-keratinized vagina. Stratified tongue. That's A. Keratinized tongue. That's B. Non-keratinized skin. That's C. Keratinized skin. That's D. Non-keratinized vagina. E. Let's have it. Let's have it. Let's have it. Let's have it. Keep going. Keep going. Okay. You guys are really active and engaged. I'm loving it. I am loving it. Perfect. You are correct. Non-keratinized vagina. All right? Because keratinization, you will never see it inside the body. And please remember, I told you that for the vagina. Okay, let me ask you this question, this sub question. The vagina is usually cuboidal, correct? Cuboidal epithelium. It now changes to what? To non-keratinized stratified squamous. What is that process of change? That change, what is it called? Please put in your answer. Let's go. Let me see if you are following me. What is it called? What is it called? I need 10 people to make attempts on that answer. Because they can ask you that question based on vagina. They can ask you that question based on a smoker. When smokers smoke, their epithelium is usually what? Pseudostratified epithelium that has goblet cells, that helps to trap mucus, or rather that produces mucus that help to trap waste. All right? It can change from pseudostratified to what? To stratified squamous. So there's no longer goblet cells to produce mucus there. You see them coughing a lot, coughing a lot, coughing a lot. What is it called? Come on. Okay. I have up to 10 people. Perfect. You guys are extremely correct. It is metaplasia. All right? Thank you for that. It shows that you guys are active and engaged. I love that. Okay, let me try you guys now. Let me test. You people have been praising you that you are so brilliant in so far. Everybody here on the live is very brilliant. Let me see. Let me test your mettle now with this question. Let me test you guys. I will not lead into temptation, but let me just test you. No option. No option. Let's go there. No option. No option. Let's go there. No option. Let's go there. No option. Let's go there. Please. Somebody said simple. Simple is not an epithelium. Oh, please. You have to give the epithelium properly. Should I give options? Yes. Okay. Stratified squamous, stratified cuboidal, simple cuboidal, simple columnar, pseudostratified columnar. Answer. Let's go. Let's go. Let's go. Let's go. Nobody is sleeping. Wake up your neighbor. Let's go. Move it. Move it. Move it. Move it. Okay. Good. We have a lot of people attempting it. The answer is simple columnar. Perfect. Like, remember I told you it's very difficult for you to see a stratified columnar. So it's simple columnar. You're good to go. Like, let's not waste too much time on it. This is the one. No, not pseudostratified, please. Not pseudostratified. Not pseudostratified. The epithelium whose cells are tall and narrow, with each touching the basement membrane. That's where you take out pseudostratified. You understand? Each touching the basement membrane and reaching the apical surface. So all of them touch the basement membrane and they reach the apical surface. All right? That's simple columnar. But if it is pseudostratified columnar, some of them will be short like this. Then some will be tall, standing on the short ones. So it will make you start thinking that, oh, maybe this is stratified, but it is not actually stratified. That is when you talk about pseudo-stratified. All right? Pseudo is false. False stratification. It's not actually stratified, but it is trying to confuse you that it is stratified. That's why we call it pseudo-stratified. And the only place you usually see it in the body is what? In the lungs, trachea. If you just hear it, please, pseudo-stratified. And don't mistake it with alveoli. Alveoli is simple squamous. Okay, I'm giving you this question now. Please, if you have any question, please put it in the comment section. right now, right here. Let's deal with it, all right? Don't be shy to ask question. No matter the country where you are currently, ask question, all right? Yeah, the answer is not pseudostratified, it's simple columnar. Okay, there are two options here, all right? This question says, dash epithelium is the most widespread epithelium in the body, whereas dash epithelium is a rare epithelium. Should I give options? If you want me to give options, click the like button there. Click it. Then I will give you options. We have up to 35 people here. So I need all of you to click the like button. Okay, somebody click the like button. Okay, so you need options. A, there are two options, right? Okay, people are clicking the love button. Okay, A, simple columnar, stratified columnar. B, stratified squamous. A, simple columnar and stratified columnar. B, stratified squamous and stratified columnar. C, pseudostratified and stratified squamous. D, simple cuboidal and stratified squamous. E, stratified squamous and simple squamous. Oh, you're welcome. All right. Oh, you'll not be here for the skin. Sorry. I really need to cover the tissue before I even jump into skin. It's fine. You can look at the video tomorrow because I know it's automatically recording. All right. Thank you so much. Don't forget to click like and follow the page. Subscribe rather. Okay, so let's go. What's the answer, guys? Come on, I need 20 people to give me an answer. 20 at least. We have up to 30 something people here. 20 people at least. Even if you are not sure, at least pick an answer for yourself, all right? It's fine. If you get it wrong, you will get correction. Dash epithelium is the most widespread epithelium in the body. Think about it. What is the most widespread stuff in your body? That is your skin. Simple. It's your skin. What epithelium is the skin? Stratified squamous epithelium. You got that answer. Immediately you see stratified squamous, you remove every other thing. You have stratified squamous and columnar, stratified squamous and simple squamous. So let's see which of them is correct. The second one says, which one is the rarest? Which one is very rare? Stratified columnar, yes. So your answer is B. You guys are wonderful. So it's stratified squamous, the most common, stratified columnar, the rarest, all right? It means you guys are really following. I love it. I love it. So let's go to the harder questions. Let's go to the harder questions, all right? Let me see you guys metal. The harder questions. Now, these are, I'll be faster now, please. I'll be faster because I know a lot of people are already going to sleep. Before we have almost 39 people now. We have 22. So people are going to sleep. It's fine. It's fine. It's fine. I need to enter the depth of this thing. A brush border of microvilli is found in dash epithelium, which can be found in dash. Option A, pseudostratified nasal cavity. B, simple cuboidal esophagus. C, simple columnar small intestine. D, stratified squamous anal canal. E, stratified squamous esophagus. Come on, let's go there. We have to 30 people here. So put it for me. Let's go. The answer. Pseudostratified nasal cavity A. Simple cuboidal esophagus B. Simple columnar small intestine C. Stratified squamous anal canal D. Stratified squamous esophagus. Answer. Okay, keep going. I need up to 15 people to make attempts. Keep going. Let's go. I hope you are not sleeping. If you are sleeping, wake up yourself. Come on, let's go. Hit that button and type something. Come on. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I need two more people to tell me the answer. Come on. See? You guys are correct. The answer is C. Brush border of microvilli is found in the intestine, the small intestine. And the small intestine is what? Simple columnar epithelium. Chicken, huh? Simple. And you are good to go. Remember, this brush border, right, is where you see some of these enzymes, right? The enzymes that break down things to their least units. That's where you find them, in the brush border. And that's what that question is asking you. It's getting a little technical. Brace up for it. Brace up for it. It's getting a little technical. Now, look at this question. Look at this question. Brace up for it. Are you there? Okay, answer. It's an epithelium. What is a particular epithelium? I need you to give me the answer point blank. I'm not giving options. Sorry. No options. Sorry. It's getting harder. I know. I'm sorry. I can't help it. I wish I can. It's fine. You can run away, but please, I need the answer. Come on, guys. Okay. Somebody's already saying something. Somebody's already saying something. Somebody's already saying something. Okay. Keep going. Keep going. Keep going. Wow. What is delaying the answer? Is it because I did not give options? Should I give options? A, pseudostratified columnar. B, simple columnar. C, stratified columnar. D, stratified cuboidal. E, stratified squamous. Answer. Come on, guys. Come on. Let's make it faster. We're up to 29 here. I need up to 10 to 15 people to give me the answers. I hope you are not sleeping. Come on. I need us to be faster because we still have some major. All these ones are just testing questions, testing the mic kind of questions. Are you thinking about it? Yes. Correct. You guys are so correct. The answer is pseudostratified what columnar. If your answer is not pseudostratified columnar, please correct it to pseudostratified columnar. All right? Because if you look at the question says all cells in the dash epithelium reaches the basement membrane. Everything reaches the basement membrane. All right? Because if it doesn't reach the basement membrane, it becomes stratified. So all of them get to the basement membrane. All right? No, it's not. It's not E. E is stratified squamous. The answer is A, pseudostratified columnar. All of them reaches the basement membrane, all right? But not all of them get to the apical surface. That's why it says, but only cells that reach the free surface have cilia, meaning that there are the ones that didn't reach the free surface. But if it is a simple columnar, all of them touches the epithelium and all of them reaches the surface. So they are just like this. But pseudostratified will make you feel that it is stratified, but it's not really stratified. It's as simple as that. I've explained it before. So I need you to look at this question very fast. Look at this question very fast. I will post the options. Please don't pick an answer until I finish posting the answers. I'm posting 5 answers for you guys. So this is, then you tell me if it's A, B, or C, or D, or whatever that is your answer. This is A. This is A. This is yeah. This is B. This is C. This is D. And this is E. Incoming missile. Incoming missile. Somebody's okay. So that's A, B, C, D, E. What's your answer? Which one is found in the urinary bladder? Found in the urinary bladder? Dash resembles dash, but the apical cells are rounded and not flattened. Answer. Wow, you guys are so hot. Like, you guys are hotting. The heat coming out from your brain is even affecting me right now. Okay. Come on, let's go there. Come on, let's go there. One, two, three, 4, 5, 6, 7. Come on, I need 10 people to give me an answer before I move. Any question I ask, I need at least 10 people to give me an answer before I move. We have 9. Come on. One more. Let's go. One more. Let's go. One more. One more. Let's go. One more. Let's go. Let's go. Yes, you are correct. In the bladder, we have the urothelium. All right. That's the transitional. It resembles what? The stratified squamous epithelium. Why? Because it is, if you look at it, it looks like a stratified. It's actually stratified, but it's not stratified squamous. You understand? So that's the urothelium, or the transitional epithelium. All right. Very, very important that you know this. I hope you guys are not feeling sleepy. Please don't sleep on me. I have very much important questions coming out. I'm trying to be as fast as possible. Please give me the answer to this very fast. Give me the answer to this very fast. Simple columnar and pseudostratified columnar epithelial have dash cells which produce a protective mucous coating. I'm not giving you option. Come on. What is contained in pseudostratified and columnar that gives out mucus? What's the cell? What's the cell? Come on. Come on. Let's see. Let me see if there are 10 people that knows this cell. Let me see if there are 10 people that knows this cell. Come on. Perfect. You guys are wonderful. Like, I am proud of each and every one of you from wherever you are right now. Okay, so that's good. Look at this question. Actually, it's goblet cells. Yeah, you know goblet cells. I've mentioned it a lot of times. It's found. That's why smokers they cough a lot, because like when in the lungs, you understand, the pseudostratified epithelium has the goblet cells. So when dust particles enters your nose, the goblet cells produces mucus and traps it. So you don't even know, you don't react. That's your first line of defense. But if it overwhelms your mucus, right, you start coughing, right? If it passes that mucus level, you start coughing. Okay, I'm coming. Let me answer you. So, but in smokers, it changes and becomes what? Stratified squamous. So it doesn't have that mucus goblet cell anymore. So if dust particles enters their nose, there is no mucus to trap it. So the person starts coughing, which is the second line of action. All right. So that's that for goblet cells. Yeah, somebody is asking, says, Professor, if the vagina can stretch during birth, like the bladder, when it's full, so why is it considered non-keratinized squamous, stratified squamous, not transitional? Yes, the thing is, it can stretch to some extent. Do you understand? But it doesn't stretch like the bladder. Trust me. It can stretch to some extent because of how it folds. The truth of the matter is that the vagina stretches because of how it folds, but not because of the cells of the area. It's just because of how it folds. It's a lot of mass. You understand? A lot of mass. So when the baby is coming, the mass starts separating. You got it? Good. Thank you for getting it. So, but the bladder, if you see the bladder is just like this, very small. But immediately the fluid starts entering it, it starts stretching. So there's a difference between stretching and expanding. All right? Stretching is stretches the cells, but expansion is maybe something that is big, opening up. It's just like I fold my clothes. You understand? Then I open it up. It didn't mean that the clothes stretched. Perfect. 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 Thank you for that question. Thank you. Extremely important question. I appreciate your question. All right. Ask as much question as possible. Sorry, that thing is abrasion. That thing is abrasion. Dash is associated with rapid transport of substances through a membrane, whereas dash is associated with resistance to abrasion. Let me give you the options. I'll post it in the chat. A, Just give me a minute. Let me post the options. This is A. Then this is B. Hold on. Then you tell me which one it is. This is B. Then this is C. Then this is D. Then this is E. Okay, let's go there. Let's go there. Which one is associated with rapid transport of substances through the membrane? And which one is associated with resistance to abrasion? Come on, guys. I need you to, fire. We're up to 30 people here. So give me answers. Give me answers. Come on, give me answers. I love when you give me answers. Come on. Only 5 people? Nah, come on. Come on. Okay, I have up to 10 people. Perfect. You guys are perfect. The answer is pseudostratified and simple squamous. Perfect. Okay. Look at this one. Okay, let me see how fast you guys can give me the answer. I'm not giving option. Each individual cell or each individual osteocyte and chondrocyte resides within a little space called dash. I told you something about sites. There's something I mentioned about sites. Anytime you see sites, there has to be something you should remember. What did I say you should remember? That's the answer to this question. Let me see if you guys were actually listening. Let me see if 10 people can remember it. Come on. Let's go. Fire it up. Perfect. Lacuna. Perfect. Yeah, you're correct. Thank you so much. Now, I want to put this option here. Yes, perfect. You guys are right. Now, I'm going to put this answer here, and I need you guys to you give me just a second to post the options, because I really need to give you options in this particular one. All right. Oh, this is that's the question. So I need you to be looking at the question and be thinking about it. Yeah, Lacuna, you're correct. This is A. This is B. This is C. This is D. And this is E. Okay, so let's go. Collagenous fibers are very abundant in dash. A, blood and hyaline cartilage. B, blood, adipose tissue, and osseous tissue. C, ligaments, bones, and most superficial portions of the skin. D, tendons, ligaments, and the deeper portion of the skin. E, areolar tissue, tongue, and bones. Where can you find a lot of collagenous fibers? Where are they abundant? Come on, guys. Let's go. Hit it up. Only 2 people making attempts. What's happening? Where are the entire 28 people? Are you running away? Don't run away from your responsibility. Come on, guys. Come on. Fed, I see you. Come on, guys. Come on. Hit it up. I need everybody to be active and engaged. Come on, hit it up. Okay. Yes, I have up to 10 people now. Good. Yes, the answer is tendons, ligaments, and deeper tissue portions of the skin. You're correct. That's D. You're correct. That's D. I'm not going to explain it if you, as far as all of you understand it. Yes, perfect. So I need you to give me this answer. I have to give you option for you to be able to get this answer. It says, which of the following does not account for the gelatinous consistency of connective tissue and ground substances? Answer A, option. Listen, please pay attention to the options. All right, once. I'll read it once. A, glycosamine glycans, that's GAGs. B, proteoglycans. C, chondroitin sulfate. D, hyaluronic acid. And E, collagen. Which of the following does not? Answer. Answer. Is it tough? Is this one tougher? Is this one tougher? Option: glycosaminoglycans, that's GAGs, proteoglycans, chondroitin sulfate, hyaluronic acid, collagen. Which one is not? Let's go, let's go, let's go. Somebody said E or D. E or D. No, you better choose one. Okay, yeah, you said E. Okay, okay, let's see. What is E again? Somebody, okay, E is collagen. Okay. Come on, where is everybody? Don't run away from your responsibility. Yes, let's go there, guys. I'm loving it that you guys are really active and engaged, trying to sort out this. All right. Yeah, this serves as your revision for your exam. Perfect. Yeah, think and answer, think and answer. Good. Yes, I get up to 10 people, 10 responses. You are correct. The answer is collagen. Remember, I told you consistency in terms of gelatinous materials. We are talking about glycosaminoglycans. Glycosaminoglycans, what makes it up? You understand? You talk about the chondroitin sulfate. You talk about the hyaluronic acid. Extremely important. All right. Now you have the proteoglycans, but there is nothing like collagen in terms of consistency. Collagen makes it hard instead of soft. All right. So that's the ideology of that question. All right. So it's nice that you guys are able to attempt it. So let's answer another one on glycosaminoglycans. Tell me the answer to this. Oh, no, I need to put options for you in the dashboard. So please just hold on. Let me give you the options. This is A. This is B here. Please bear with me. I know I'm a little slower. Still getting the grips of this stuff. This is C. This is D. Incoming missile. This is D. And this is E. Okay, so let's go. Glycosaminoglycans. Sorry. Glycosaminoglycans, that's GAGs, help give the ground substance some of its qualities, including what? Let's go there. Let's go there. Keep going. Keep going. Keep going. Okay, you guys are correct. It helps, including holding water and maintaining electrolyte balance. You know, I discussed it when I was discussing it in the slide. Oh, perfect. Perfect. This is the kind of questions that you definitely will see in the clinic. Thank you, guys. So let's try this. Let's try this. Let's try this. I'll give you the option. Yeah, you guys are still trying the other one. Yes, the answer is B, holding water and maintaining electrolyte balance. Thumbs up, thumbs up. You guys are doing great. Now, let's talk about this. An example of a highly vascular tissue is dash tissue. An example of a tissue that is not highly vascular is dash tissue. A, epithelial cartilage. B, muscular osseous tissue. C, dense regular connective, dense irregular connective. D, areolar cartilage. E, nervous tissue, osseous tissue. Answer. People that have answered before, you know you need two answers, not one. A, epithelial cartilage. B, muscular osseous tissue. C, dense regular connective, dense irregular connective tissue. E, areola and cartilage. F, Abi, the last one, nervous and osseous tissue. D is areola cartilage. Then E is nervous and osseous. Okay. Somebody said C. C is dense regular connective and dense irregular connective. B is muscular and osseous. A, epithelial and cartilage. D, areola and cartilage. E, nervous and osseous. Which one is it? Keep going. Somebody said D and E. Keep going. Okay, you removed your E and you choose D. That's good. Let's keep going. Yes, the answer is areola and cartilage. You remember I told you areola is very highly vascularized. That's why what you usually see, that's D. Yes, you guys are correct. You guys are very, very great. So areola is the connective tissue you see under the epithelium. Remember, the epithelium is avascular. It has no blood supply. It's a vampire. It needs to get blood from someone. Who is it? The connective tissue below it. And that connective tissue is supplying itself and the people above it. All right? So because of that connective tissue needs to be the most highly vascularized connective tissue. Which one is it? Areola. And cartilage is the one that is not even vascularized. All right? It's not. In as much as the connective tissue is still not vascularized. All right? Now, I'm going to put this question, and please, I'll read the options. And we are almost about to end for tissues. For people that are strong, you can continue with me for skin. Now, look at this question. Which of the following is not a feature that connective tissues have in common? Which of the following is not? Whenever you're answering question, always look for the caveats, all right? Extremely important. Option A, very conspicuous fibers. Option B, presence of fibroblasts. Option C, the presence of collagen. Option D, ground substance with gelatinous to rubbery consistency. Option E, a calcified matrix. Which one is not a feature of connective tissue? Which one is not a feature of connective tissue? Do you still remember the features of connective tissue? These are the ground substances that we are talking about. They are part of the features. These are the fibers, the collagenous, the reticular, and the elastic. These are the cells. These are the functions. And these are other things that they do, all right? Like that, this is the cartilage I was talking about some minutes ago. Cartilage has few or no blood cells. Why the loose connective tissues are the ones that has the major blood supply, like what? Areola, adipose, and reticular. All right? So tell me, what's the answer to my question? Which of these is not a feature that the connective tissue have in common? Very conspicuous fiber is A. The presence of fibroblasts is B. The presence of collagen is C. Ground substance with a gelatinous to rubbery consistency is D. A calcified matrix. What's the answer, guys? Come on, I'm seeing only 5 people out of almost 30. Come on, please. Please, the questions are getting interesting. And I will be extremely fast. So the faster you answer, the better for me to move on, all right? I need to be sure that you understand what we are discussing. Okay, yes, you're correct. The answer is E. That's a calcified matrix. We don't care about that. You don't discuss that in the connective tissue. The shape of a person's ear is due to the presence of dash. A, dense regular connective tissue. B, Dense irregular connective tissue, C. Elastic cartilage, D. D. Fibrocartilage, and E. Ligaments. Answer. Let's go, guys. If I get 5 people to answer, I move. I won't wait for 10 people anymore. I won't wait for 10 people anymore. Come on. Let's go. Don't be tired. Please don't tire out on me. Don't tire out on me, please. What's your answer, guys? Okay, so we have a lot of people. What type of connective tissue? Let me see. What's the question again? The shape of a person's ear, yes, it's elastic cartilage. Yeah, you're correct. The answer is C, elastic cartilage. The answer is C, elastic cartilage. So give me this very fast. Give me this. Nervous tissue. We are almost at the end of tissues. Almost at the end of tissues. Nervous tissue consists predominantly of two cell types, neurons and dash. Answer. Very fast. Options. Do you need options? Sorry. We have fibroblasts, we have chondrocytes, we have neuroglia, we have the myocytes, we have the osteocytes. Remember when I was discussing the nervous tissue, I told you that the nervous tissue is made up of two major things, dash and dash. Neurons, we already discussed neurons. There are something that is protecting the neurons. What is it called, guys? Come on. 5 people give me the answer and I move. Keep going. Keep going. Yes, the answer is neuroglia. It's neurons and neuroglia. Extremely important. Okay, we are coming to the end. Tell me the answer to this. Neurons usually have a multiple short branch processes called dash, which receive signals from other cells and conduct messages. You understand? True. Conduct messages to the dash. Options. A. Nerve fibers, dendrites. Dendrites, cell body. Axons, dendrites. Neuroglia, cell body. Nerve cells, glia cells. Keep going. Everybody has to be engaged. I will ask you. I'll ask you 3 questions. Okay, four more questions. 4 more questions to go. 4 more questions to go. 4 more questions to go. Are you ready? Are you ready? Yes, you're ready. Okay, thank you. Yes, the answer is dendrites and cell body. You're correct. I'm trying to hurry up. So this is the question. I'll give you options. Hold on, let me post the options in the group. I'll give options. A. I have 4 more questions to go. So for tissues, so just hold on. Even if you want to go, you can go once I'm done with tissues. Please click the like button, guys. I want to see that like button smiling. That's C. This is C. Then this is D. This is D. Yes, keep liking. Keep clicking like, man. Guys, keep clicking like. This is the last one, E. Okay, guys, give me the answer quickly. Answer. Let's go there. Let's go there, guys. Okay. Yes, endocrine glands secrete hormones into the blood. Please, this is a very, very interesting question. All right. This is a very, very interesting question. So they may try to confuse you with this question. Now, this is I want to post one of the most beautiful questions that I'm very, very sure. I left some of these interesting questions for the last, especially for people that will be here till the end. So, so that they will be like, oh, wow. Let me see. Just hold on. I want to post this. Now, I'll post this question. I need you to calm down and look at it. I'm posting this question. I need you to take your time to look at this question. Guys, this question is not just any question. Trust me. We are now going clinical. I'll give you the options. This is A. This is as expected. We reserve the best for the last. This is C. This is D. And the final question. The final option, rather. This is E. Please think well. This is one of the most important questions you are most likely going to see in your exams. I want you to think well, then I will show you something from the slide. So think well and give me the answer. Come on, guys. Let's go. Let's go. More people to comment on this. This is one of the most important. So I need everybody. We have more than 20 people here. I need you guys to attempt it. Even if you don't understand it, you can write "don't understand" in the comment section. It's fine. I will explain. If you don't understand, write "don't understand." Keep quiet, please. I really need to make sure that all of you are active and engaged. All right? Don't slip up on me, please. Please. Come on, guys. Come on. If you are not sure of the answer, just write "not sure." It's okay. Okay, that's fine. So if you look at this, I want you to look at this slide. Gap junctions. Remember what I said gap junctions are? Gap junctions are communicating junctions, right? Remember we have tight junctions like this. Tight junctions. Yeah, thank you. I will explain. So we have good, perfect. I like anytime you are not sure of an answer, don't just keep quiet. Don't follow others. Right? Not sure or explain. I will explain, all right? So that I'll carry along because you are awake by this time. You have to have something to show for it. You have to go and sleep after this discussion and dream happily. All right? Tomorrow you come to the exam with swag. This is tight junction. This is gap junction. The connecting things there is the desmosomes. What you are standing on, what the cells is standing on is what? The hemidesmosomes. Now, that gap junction is not found everywhere, right? Look at this page. You will see connexons consist of 6 transmembrane proteins arranged like segments of an orange around water-filled pore. Ions, nutrients, and other small solids pass through. But look at the most important thing I need you to understand. Where is gap junction found? Gap junction is found in the cardiac and the smooth muscle. Remember, gap junction is found where? Cardiac and smooth muscle. Embryonic tissue, lens, and cornea. Did you see skeletal tissue, muscle? No, it is not found in skeletal muscle. Instead, it is found in what? Cardiac and smooth muscle. Meaning that if there is a problem with somebody's gap junction, that person will be having what? Heart problem and smooth muscle problem. But the person will not have any problem with skeletal system, because why would you have a problem with skeletal system since there is no gap junction in the skeletal system? So the only problem you'll be having will be places where gap junctions are found, like where? Like the cardiac and the smooth muscle. So if you go back to that question, you will see, hold on. So if you go back to that question, they say, how would the genetic inheritance of a gene producing abnormal gap junction? So they are trying to tell you, oh, this gap junction is terrible. The gap junctions this person has is bad. Where will be affected? So the answer will be the places that are affected are the places where you can find gap junctions. Like where? Look at the option C. Option C says cardiac muscle, smooth muscle. Yes, they will function abnormally because gap junction is found there. All right. Other places that will function abnormally is the lens, because gap junction is also found on the lens. Now, that C says skeletal muscles would be unaffected. Of course, they will be unaffected because there is no gap junction in the skeletal muscle. So there are people that say they are not sure, they don't understand. Please, if you understand now, right? Yes, I understand. If you don't understand, I can explain more. Perfect. Now there is this because you guys have been here, so I want to reward you with this very simple question. A, striated and voluntary. B, striated and involuntary. C, striated, C is non-striated and voluntary. D is non-striated and involuntary. E is fibrous and containing ground substance. Answer. Answer. Yes, you are correct. Striated. Yeah, striated and voluntary. Now, look at this subclinical question. You understand? Look at this subclinical question that looks like the things that they... Okay, you said I should repeat. Yeah, of course, I can. Not a problem. I said skeletal muscle is described as dash. A, striated and voluntary. B, striated and involuntary. C, non-striated and involuntary. D, non-striated and involuntary. And E, fibrous and containing ground substance. And yes, the answer is A, that's striated and voluntary. Voluntary skeletal muscles are striated. What causes their striations? Alternative what? Light and dark bands. That's what causes their striations. I need you to remember that. Alternative light and dark bands. And it is what? Voluntary. Cardiac muscles are striated but involuntary. And what causes the striations of cardiac muscle? The intercalated disc. Smooth muscles are not striated and what? Involuntary. Look at this question, guys. Take your time. Don't rush. You're welcome. Don't rush. I'm putting the options in the box. Don't rush it. Take your time. This is A. This is B. This is D. And this is E. Oh, it's freezing? Oh. Are you in New York? I don't know it's freezing, honestly. Man, this weather has changed, man. So what's the answer? Astronauts in zero gravity are able to move food through their digestive tracts because dash. A, smooth muscle produces wave of contraction that propel material through the digestive tract. B, skeletal muscle puts pressure on the digestive tract. C, cardiac muscle contains a high pressure that moves material through the digestive tract. D, striated muscles creates a pressure gradient that forces material from one end of the digestive tract to the other. And F, skeletal muscles, sphincters contract and allow materials. Okay. Wow. So it's freezing in New York already. Hi, God, though, please. I really love the hot weather all along. Okay, come on, guys. Come on, let's go. Come on, let's go. What is the answer? Come on, guys. Pour in the answer. Pour in the answer for me. Let me move. If I need 10 people, just 10 people for me to move. Come on. I need 10 people to move. Yes, the answer is A. The answer is A. I'm not going to discuss it. So, please, I'll just say some answers very fast. Oh, the screen is freezing. Oh, I thought you were talking about the weather. Am I freezing? Oh, sorry. I don't know. I thought you said the weather. Okay, you don't understand. Yeah, so the question, hold on. The question says astronauts in gravity are able to move food through their. What they are just asking you is to know if you understand that in the digestive tract you have smooth muscle. That's what they just want to ask you. And that's why the A is answer. A says smooth muscle produces waves of contraction that propel material through the digestive tract. Of course. Skeletal muscle puts pressure on the digestive tract. It cannot put pressure because you are in space. Cardiac muscle maintains a high pressure that moves material through the digestive tract. Cardiac muscle is in the heart. It has nothing to do with the digestive system. Striated muscle creates a pressure gradient that forces material from one end of the digestive tract to the other. It's not true because the smooth muscle in the digestive system is not striated. Skeletal muscle sphincters contract and allow materials to move through the digestive. It is not the sphincter that causes contraction. It's not. It has nothing to do with contraction. Contraction is just the skeletal, the smooth muscle peristaltic movements. All right. That's why A is the answer. So smooth muscle produces waves of contraction. All right. Good. Okay. Good. Okay. So guys, I want to end the tissues, but there are still questions I need you to know. So I'll just discuss the questions very briefly. Let me jump to smooth to skin. There's a question they said in the intestine, dash ensures that the most digestive nutrients pass through the epithelial cells and not between them. No, I think I need you guys to attempt this question, please. Very fast, very fast. Then I'll give you the answer. Because I think this question sounds very important and clinical. Most of you are in the clinic. So in the intestine, dash ensures that most digestive nutrients pass through the epithelial cells and not between them. A, plasma membrane. B, desmosomes. C, tight junctions. D, gap junctions. Then E, ground substance. Come on. I need 5 people. Come on. Let's move. Don't get tired of me, please. 2 more. Perfect. Yes, the answer is tight junction. Thank you so much. The answer is tight junction. So the last, let me see. Oh, this question is too long. And I really need you guys to attempt this question. Let me see if I can break it down. Yes, you're correct. Tight junction. Oh, we still have a lot of people here, up to 27 people. Perfect. I really like you guys. You guys are so resilient, man. We've been together for a lot of hours. You guys are resilient. I love it. I am loving it. There is this question I want to ask. Hold on, I'm trying to reduce the question. Dash glands. Okay. Hold on, I'm trying to put in the question in the box. This question is too long, and it's refusing to enter. I don't know how to be able to put this question now. Still have almost okay. Okay, perfect. Okay, so guys, let's attempt this question very fast before we run out of this place. Dash glands, such as tear glands, have vesicles that release their secretion via exocytosis. Dash glands, such as oil-producing glands, secrete a mixture of disintegrated cells and their products. Answer. Okay, I'll give you options. A, please pay attention to the option. Yeah. Option A, endocrine, exocrine. B, mucous serous. C, exocrine, apocrine. D, mucous cutaneous. E, ecrine, holocrine. Please, if you don't know the answer, right, don't know. All right, no shame in it, so that I can explain. Come on, let's go. Only one person. Come on, let's go. Move it. I want to jump to skin ASAP. Okay. I like the fact that people are attempting different things. Okay, I'll give you the answer fast. So look at this diagram. Look at this page, all right, so that you'll be able to see the answer. What did we say? We say merocrine secretion, which is ecrine glands. What are the examples? Tear, pancreas, gastric glands, and others. Apocrine, milk. All right, that's apo, lipid. Holocrine is what? Oil glands of the scalp and skin. So what is the question again? The question says dash glands such as tear glands. So example of tear gland is what? Ecrine. Then the second part says glands such as oil producing glands, which are oil producing glands, holocrine. So the answer is E, ecrine and holocrine. All right. Okay, guys. So I think we will leave it at that for this guy. So please, I need you to note the hypertrophy and hyperplasia. All right, very, very important. Hyperplasia and hypertrophy, extremely important. You need to know that. You need to know what metaplasia is. You need to know atrophy. You need to do differentiation. You need to know neoplasia. You need to know necrosis. You need to know apoptosis. All right, apoptosis is programmed cell death. These things you need to know. Please, if you have any question, put it in the comment section. I'm going to take 10 minutes or 15 minutes to discuss little information on the skin before we call it a day. All right. For people that can, you can remain. But if you are too tired, it's fine. You can go. You understand? A lot of professors don't set so much questions on skin, but it's still good if you can pay attention for the next 30 minutes. We'll be done with this. All right. So this is the skin. All right. Study is dermatology. Extremely important that you know that. This is the architecture of the skin. We have the epidermis, we have the dermis, we have the hypodermis. All right. Epidermis, remember, there is no blood circulation on the epidermis. If you look at it, you will not see any vessel because it's avascularized. That's the epithelium of the skin. But this is the connective tissue. You see how vascularized it is? Very, very important. And this is fat tissue. All right? That's the cutaneous tissue. Like if you want to give injection, you give the injection under this. We call it subcutaneous. All right? These are the layers. We have the epidermis, dermis, and the hypodermis. Very, very important. And also, you have the thick and the thin skin. The thin skin, you see it everywhere, but the thick skin, you see it on the hands and the bottom of the feet. All right? Extremely important. And it's very, very thick, extremely thick. So these are the things, the functions of the skin. Extremely important that you know it. These are the functions. Then we have the epidermis. I told you that the epidermis is the keratinized stratified squamous epithelium. And remember, it has no blood supply, it lacks blood supply. So it's supplied by the connective tissue, that's the areolar connective tissue. Also, you need to know the cells of the skin, the cells of the epidermis specifically. We have the stem cells, we have the keratinocytes, we have the melanocytes. And you need to know where they are found. All right? So then we have the tactile, we have the dendritic cells as well. Okay, so these are the layers of the skin: conium, lucidium, granulosum, spinosum, and basal. These are the 5 layers of what? The epidermis. Then we have the dermis here. All right, and others. So I don't want to continue this because there are a whole lot of things you can see from here, but I just want to give you 10 questions only, and we call it a day. I want to give you 10 questions only, and we call it a day. Are you ready? So please remember the friction ridge and flexion line. All right. Flexion line is the crease. Friction ridge is the one that causes your fingerprints. Very, very important for you to know. So I won't continue discussing this slide. All right, because I believe that most of you already has the slide. So I want to give you just 10 questions. Just 10 questions. Only 10 questions. Only 10 questions from this, and we call it a day. I'm also tired. Okay. Okay. So I want you to see this. So for people, I need just 5 people to make a comment on this. For people that are still here, let's go. Let's fire it. Which of the following is not found in the epidermis? We have the melanocytes, A. B is the fibroblasts. C is the stem cells. D is the keratinocytes. E is the tactile cells. Answer. Answer. Come on, let's go. What is the answer? The cells of the epidermis. I'm waiting. A is the melanocytes, B is the fibroblasts, C is the stem cells, D is the keratinocytes, E is the tactile cells. Which of the following is not found? I need 2 more people to comment. We are 20-something people here. 2 more people. Come on, let's go. Let's go. Let's go. 10 questions. 10 questions. Go in. Go in. Okay, good. So fibroblast is not. If you look at the option, the slide here, you see 5 epidermal cells. We have the stem cells, we have the keratinocytes, we have the melanocytes, we have the tactile cells, and we have the dendritic cells. You will not see fibroblast there. All right? You won't see fibroblast. So that's a very, very important thing. Questions you need to remember. So let's go clinical. Number 2 question. I told you 10. So just bear with me. Let's discuss 10 questions and you can go. All right. Wow. Immediately I started the question. Everybody started rushing in. That's good. That's good. That's good. In which of the following regions would a subcutaneous injection be administered? For some of you that give all of you are nurses. So for you guys that give subcutaneous injections in the clinic, where do you put the injection? Is it in the stratum basal? Is it A? Is it in the stratum spinosum? B. Is it the stratum corneum? C. Is it in the dermis? D. Is it in the hypodermis? Okay, somebody say hypodermis. Okay, keep it coming. Keep it coming. Keep it coming. Keep it coming. Okay. I think that's up to 5. The answer is hypodermis. You're correct. Okay, so look at this question. Please keep count. Dendritic cell A. We have the melanocytes C. We have the tactile cell D. We have the dead keratinocytes E. Let's go. 2 people have attempted. Keep it coming. Correct. The answer is melanocytes. You guys are very correct. I hope my screen is not freezing. Oh, sorry. I know it's freezing a little, but please just bear with me, all right? Look at this one. The fastest rate of mitosis happens in the A, stratum basal, B, stratum corneum. Which part? The fastest rate of mitosis, where does it happen? Stratum basal, stratum spinosum, stratum granulosum, stratum lucidium, stratum corneum. Answer. Perfect. Perfect. You guys are correct. It's basal. That's the basic layer. It's basal, all right? It's basal should be A, yeah. It's A. I mentioned it as A. So please note that, all right? So give me the answer to this very fast. I'm trying to hurry up so that I can leave you guys. I know a lot of people are already dozing. Which layer is seen only in the thick skin? Stratum basal, stratum spinosum, stratum granulosum, stratum lucidium, stratum corneum. Answer. A, basal, B, spinosum, C, granulosum, D, lucidium, E, corneum. Answer. Which layer is only seen in the thick skin? Like the palm of the hand? One more person. One more person. Okay, lucidium. You're correct. Okay, look at this question. The hypodermis is characterized by the abundance of which tissue? Which tissue do we have more in the hypodermis? Okay. I hope, please, if I get up to 10 questions in skin, tell me so that we can stop there. The hypodermis is characterized by the abundance of which tissue? Let me see 3 people that can get this. Okay. Keep going, keep going, keep going, keep going, keep going. We have 3 people. Let me get 2 more. Keep going. Perfect. So adipose tissue is contained a whole lot of skin discoloration most likely to suggest physical abuse is what? Let me give you option. We have A, pallor. Okay, 4 more. Just tell me when I'm done. A, when is 1 more? Tell me. We have A, pallor, B, albinism, C, erythema, D, hematoma, E, jaundice. The skin discoloration most likely to suggest physical abuse. In the clinic, they brought me a child to the hospital. As a clinician, there's something we call reportable offense, right? You must report if you see these things. What are you looking out for children, especially? Exactly. You guys are so correct. You guys are so correct. I'm proud of you. I'm proud of you. I am very proud of you. I wanted to ask this question, but I'm not asking it because it will count to my question. Which skin color is most likely to result from anemia? Please, it's pallor. I'm not saying anything. Remember, in the clinic, you check pallor in several places, like the eyelids, lower lips, right? Capillary fill, all those places. You check. So I'm not counting that as part of my 10 questions. So I'm giving you this. Look at this. The reddish color of the skin of the lips is primarily due to the presence of which pigments? Which pigments? The reddish color of the skin of the lips is primarily due to the presence of which pigments? We have hemoglobin, we have myoglobin, we have melanin, we have carotene, we have keratin. Keep going, keep going, keep going, keep going. Hemoglobin. Perfect. The answer is hemoglobin. Perfect. You guys are the best. You guys are the best. How many questions do I have more? Here is the timekeeper. Okay. Look at this question. Look at this question. A dash is an elevated patch of melanized skin. A dash is an A, hemangioma, B, mole, C, freckle, D, flexion line, E, friction ridge. I'll go again. A dash is an elevated patch of melanized skin. A, hemangioma, B, mole, C, freckle, D, flexion line, E, friction ridge. Mole. You guys are so correct. You guys are so correct. Is this my last question? Is this my last question, guys? Let me see. If it's my last question, say yes in the box, in the comment section. Say yes. Okay, it's not my last question. Oh, one more. Okay. Oh, okay. So last question, last but not the least. Don't fail me. What is the hair cycle? Should I put options? Okay, let me put options. Let me put options. The hair cycle. I know a lot of people may not have heard this, but this is one of the most important questions out of this place. Fine. This is A. I'm posting. Just hold on. Let me post the 3 because the arrangement differs so that you'll be able to get it right. This is B. This is C. Okay. Put your answer in the comment section. Let's go. Let's go, guys. Let's go. Last question. Last question. Going. Is B. You're correct. Anagen, catagen, and telogen. Anagen, catagen, and telogen. So thank you very much all for joining my live today. I appreciate you guys, and I'm happy to be of help. If you have any problem, if you have any question, please always try to come on the channel, make comments, go to the videos that you have problem with, put comment on it. If there's something you don't understand, just give me the particular minutes or the time, and just ask the question there, make comments. Also tell your friends to look at these videos. This particular video will be on YouTube here, so you can always refer to it. All right, always refer to it. And I wish you success in your exams, both in your college exams, your NCLEX, and any exams that you are taking. Thank you so much, guys. Thank you. I appreciate. One love. Thank you. I came here and I didn't see any of you. Yeah, so I hope all of you are going to score 100 over 100 in your exams. All right. Thank you so much. Thank you. Thank you. Have a nice night. All right. Appreciate. Yes, I appreciate. I appreciate. Thank you so much. Please don't forget to follow to subscribe to the channel. Also tell your friends. Always click like in every video. Tell your friends to look at the videos on the channel, to really help them. More videos will be coming out very soon. I'll try to make more videos this week or next week. You understand? So click the notification bell so that anytime a new video comes in, you are the first to be notified. All right? Thank you so much, guys. I appreciate. Have a nice night. We have a good number. Bye-bye.